Do the intro. Action.
1 second. Yeah, you're good.
Okay. Good evening. Welcome to Antarctica. This is dive 777 of the ROV Sebastian. We're diving off the research vessel Falco 2, which belongs to the Schmidt Ocean Institute. We're here in the North Ronne Entrance, which is in the Bellinghausen Sea, off the western side of Antarctica. The objective today is to, we're going to start with some video transects, and then later we're going to start collecting corals and other macrobenthos, and then later on we're going to do a full set of push cores. Buenas tardes, bienvenido a Antártica. Eso es la emisión 777 del ROV Sebastian, que está saliendo de la buque de investigación Falco 2, que pertenecen de la Schmidt Ocean Institute. Estamos aquí en la North Ronne Entrance, que es en el mar Bellinghausen, en la costa oeste de Antártica. Vamos a empezar con algunos videotransectos y después vamos a tomar muestras de corales, otra macrofauna, y poco más tarde vamos a tomar un juego de push cores. But if you see one, come. Because you need at least 3. You need at least 3.
At least?
You need 3,
yes,
yeah. So they have to be common in.
Yes, but a médico knows very well.
¿Has visto que lo veo?
No,
Por.
sí.
Aquí.
Aquí, sí.
Otra vez.
No, es que Patricia
Sí.
me equivocó.
Seafloor, because otherwise we see mostly.
Yeah, that's okay. Thanks for that. Yeah, and what, 0.3?
Yeah, that's fine.
0.3 come speed?
Up to that is fine. Earlier today we did it 0.2 and it was okay.
Hey everybody, good evening and welcome. You just got a brief little intro from Dr. Matthew Lee, who will be narrating this portion of the dive. I hope you all are having a great evening. And right now, piloting is Andy and Joel. Thank you all for joining us. And if you have any questions, please pop them into the chat, and we will answer them for you.
Okay, we're doing 0.3 at the moment. If you want to increase the speed, we can and go up to 0.5, 0.4.
0.5 is okay, thank you.
Okay.
Well, we're just pulling away from the steadily.
Yeah. Okay. Hi. This research cruise is part of the, it's called the Climate Connections at the Sea Ice Interface. So we're obviously looking at the impact of how the retreat of the ice shelves affects the geology and the biology in this region, which is one of the parts of the planet that's changing most quickly. This research cruise is also part of the Challenger 150 series of expeditions. So Challenger is a group of deep sea biologists who are collaborating to organise expeditions in the hope of filling in the gaps that we have in our knowledge. What's that? It's an Echiourian. So yeah, what we're seeing here is a seabed that's composed primarily of very fine sediment with a high amount of organic material in it. From the ship, you can see that the water is quite green in colour, so there's a lot of primary productivity in the water column, and eventually that all sediments out, which is sort of the greedy yellow flocculent layer you can see on the surface of the sediment. And so the fauna that we have here mostly processes sediment. You can see lots of sea pigs, which are echinoderms, the group Holothuroidians, also known as sea cucumbers, but this particular transparent group are often referred to as sea pigs. We also see a lot of other echinoderms, so there's ophiuroids, we've got some sea urchins. I've also just seen an Echiourian worm, spoonworms. Some of these tubes that you can see sticking out of the sediment are probably polychaetes. And there's also quite a few fish knocking around. We've got some shrimp. And hopefully when we collect samples later, you'll find an awful lot of small worms, microscopic organisms, the meiofauna, which is what I study, in amongst the sediment, feeding on this organic material. Bueno, que puede ver es una planicie de sedimento súper fino. Aquí tenemos una alta concentración de materia orgánica. Cuando veo desde la búsqueda, puede ver el agua es verde. Es una indicación de la alta producción primaria que hay en la columna de agua aquí, y eventualmente es toda esa materia orgánica se sedimentar sobre su superficie de sedimento. Por ende, la mayoría de los organismos que se ve son procesando esa materia orgánica. Uno de los grupos más evidentes son los pepinos de mar, o en este caso de chanchito de mar, son el grupo de Holothuroides, que son equinodermos, pero también se puede ver ophiuroideos, varios tipos de erizo. Aquí estamos mirando un sipen que está pasando ahora. También hemos visto ese gusano de cucharada, que son Echiourians. También a varios puntos veo rocas. Esas rocas son rocas que son transportadas por el hielo, y cuando derrite el hielo, esas rocas caen sobre el fondo y proporcionan un sustrato duro que está colonizado por varias especies como los corales, la hydrozoa, brachiozoa. También nosotros hemos visto brachiópodos, varios tipos de poliquitos que viven en tubos, etc. Ellos están principalmente filtrando la materia orgánica que está en la columna de agua. Should introduce myself. My name's Dr. Matthew Lee. I work at the Centro IMAR in the Universidad de los Lagos in Puerto Montt in southern Chile. And I'm here on this cruise working with Dr. Americo Montiel and Dr. Cristian Aldea, who are from the Universidad de Magallanes in Punta Arenas in southern Chile. We also work with Dr. Eulogio Soto from the Universidad de Valparaíso in central Chile. And our objective as a group is to collect and study the benthos, both here in Antarctica and obviously in previous cruises with the Falco earlier this year. Last year, we were collecting pretty much along the length of the coast of Chile and also out in the seamounts out to Rapa Nui. Hola, mi nombre es Dr. Matthew Lee, soy profesor en el Centro IMAR en la Universidad de los Lagos en Puerto Montt, en el sur de Chile. Y estoy aquí a bordo con mis colegas Dr. Americo Montiel y Dr. Cristina Aldea, son de la Universidad de Magallanes en Punta Arenas, en el sur de Chile. Y también trabajamos con Dr. Eulogio Soto, que es de la Universidad de Valparaíso, en el centro de Chile. Trabajamos juntos estudiando los benthos en la costa de Chile y también en Antártica. Este año somos privilegiados de trabajar con el Falcor y muestrear en casi todas las costas de Chile. Desde Rapa Nui, la plataforma continental, desde Valparaíso hacia Punta Arenas, mirando los cañones y las emanaciones de metano que hay en el sedimento en los cañones en el sur. Y ahora estamos aquí en Antártica, mostrando un área que es poco conocida. Okay, so there's an example of a dropstone. It's passing on the right-hand side of the screen with the sponge on it. So these are stones that are transported by the ice sheets out over the ocean, and then as the ice melts, they get dropped onto the seafloor and get colonized by all kinds of sessile fauna. Entonces es un ejemplo de esas piedras que caen de hielo y son transportados desde el continente hacia el mar, y cuando derrite de hielo se caen por sobre el fondo marino, donde están colonizados por varios tipos de invertebrados sesiles. Poliquito. Puede ser. You can see around each of the ophiuroids, the brittle stars, this sort of shadow of lighter sediment. That's where the ophiuroid is processing the sediment and cleaning it of the organic material. Con los ophiuroides en superficie de sedimento se puede ver como un sombra de sedimento más liviano, más blanco, y ese es donde los bichos están procesando el sedimento, está sacando una gran proporción de la materia orgánica que hay. Eso es su alimento, obviamente.
Okay, so hi everyone, I am a master's student. I'm from Germany, from the Senckenberg Institution, and I am very blessed and feel very good gratitude that I'm here right now, participating in this Antarctic cruise. And yeah, I'm very much looking forward for all of the samples. You can already see a glimpse of what we are about to sample, looking at the bottom of the seafloor. Lots of sea cucumbers, anemones, like worms and sponges. It's beautiful out there. So I hope you enjoy.
any language you like. Yeah, come on. Do it German if you want.
German.
Yeah, why not? Don't.
Ja, das Ganze kann ich auch nochmal in Deutsch machen. Ich bin Antonia. Ich komme aus Deutschland. Ich bin vom Senckenberg-Institut hier. Ich bin eine Masterstudentin. Ich komme ursprünglich von der Universität von Oldenburg. Und ich habe das unglaubliche Privileg, Teil dieser Expedition zu sein. Jetzt gerade sind wir hier live in der Unterwasserwelt in der Antarktis. Wie man sehen könnt, ist hier sehr, sehr viel. Wir können Fische sehen, wir haben sehr viele Seegurken, zwischendurch haben wir sehr viele Schwämme, Seefedern. Es ist unglaublich viel los auf dem Meeresboden. Ja, es ist ein absolut fantastisches Erlebnis, das hier sehen zu dürfen. Wir haben hier noch einen weiteren Wissenschaftler, den wir jetzt mal vorstellen möchten. Tom, do you want to introduce yourself?
Hello everyone, I'm Dr. Tom Lindley. I'm originally from the UK, but I'm over from Te Papa Museum in New Zealand, and just feeling really lucky to be out here. I've not gotten over the Antarctic scenery just yet. Spent a lot of time staring at the horizon because it's just constantly changing, and there's this weird peace. The air is really still. Even when it's quite windy, there's this there's this vacancy to it. It's really, really haunting. So my first time down in the ROV command room, and it's a little bit overwhelming. 3 of the walls are covered in screens, and it's great to see what's going on down here. So I'm mainly a fishy person, and we've seen a few good ones come past so far. Those silvery ones that sort of tend to undulate just their tail. They sort of shimmy their tail and they keep their body quite rigid, really reflective sides. And the ones with quite a long sort of beaky snout, those are Antarctic Jacobfish, I think. But there's quite a few other things we've seen coming past. There's some sort of smelty looking fish. I haven't had a good look at one of those yet to see how many lateral lines they've got that would let us to figure out a little bit more about what they are. And I think I've also seen some plunderfish. They usually sit on the bottom, and they're a little bit like the anglerfish, but their lure comes out of their bottom lip. So on the bottom of their lip, they've got this little fleshy lure that sticks out, and apparently they sometimes eat it accidentally. Oh, wow, I can see the chat. Hello. Yeah. Anyone who recognises my voice, hello, you're special. Oh, there we go. Somebody beckoned for a Dumbo. So there's that going up the top right. That was one of the Antarctic Jacobfish. That interesting sort of shimmy.
Big sponge.
Wow, that is a huge sponge. I'm not sure if it grew upright once and then fell over. It does have a sense that it used to be the other way up. And are those some crinoids growing on it? Looks like it. What's the distance between the laser scale?
10 centimeters.
Oh, so this is big. I don't know if anyone watching heard that, but the distance between those two green dots of the laser scale is 10 centimeters. So these are some big organisms. someone on the chat was asking about the light law system that me and Kat Bolstad were putting together. So it had its first proper use last night, full 7-hour deployment, one at the about 500 meters deep and one at 200 meters deep, and hey, everything worked, which was a big relief. And I'm finding it hard to sort of bounce back. Actually, it was a full-on night, so I'm feeling a bit disorientated now after all that. I've been going through the video. That's what I was sitting in the back of this room doing before I was thrust in front of a microphone. So I haven't seen much yet, but it was a massive relief initially just to know that all the tech worked, including last minute a science sponge. I couldn't get the camera. Once you get your eye in, you can sort of see the difference in the sediment texture. So you can see the bioturbidation, or Lebensspuren, if we want to get our German in there as well, where the animals are sort of digging and feeding. So you can see those lighter areas where it's been cleaned up, and then the sort of more golden, almost getting to green areas are where material has fallen down from the surface. You can see it in the water column that there's loads of food raining down. And actually, when we were deploying our pelagic system last night, the water's surprisingly green. Something about the light here, it looks really blue and really clear from the surface, but then you actually get in the water, it's got a fair bit of photosynthesis going on, fair bit of chlorophyll in the water. So it's obviously just raining down here, and that is the food source for most of this community that we're seeing here. We're seeing mainly the cleanup crew who are gathering all that material that's raining down as marine snow. Oh, nice sea urchin on the far left. I don't know my sea urchins very well, but the ones with the really thick spines on them, they look almost like pencils. Well, they look like a blob of Play-Doh that's had a load of pencils stuck in it. That's my very scientific assessment. So at the moment we're steaming along as a transect, which is a little bit tempting. You see lots of exciting things as you go along, but basically we're trying to get a quantitative assessment of this community, basically. So we've got the laser scale on, we'll fly at a fairly consistent height and speed, and so then we can figure out the density of these animals later when we process the videos. It's tempting to sort of get sucked in by the interesting things you see and go and have a look at everything, but then that makes it hard to then process that quantitative assessment. And apparently the same thing happens with the people who pilot the Mars rovers, and they have a name for it. They call it the giant ball of twine effect, in that you're trying to go along and make progress, and you're just constantly getting distracted by road signs at the side of the road, you know, world's biggest ball of twine and tourist traps and things like that. So they use that as the analogy there. And they have to debate even more than us about how they use their time. The deep sea and space communities have started working together a lot more, actually. There's a huge amount of parallels, both in the science we do, now that it looks like any extra life in our solar system is likely to be deep sea life from one of the ocean moons, but even just how we, one of these huge sea stars. I found out what it was yesterday, but those black tips to the arms. One of the other team members showed me a picture of people holding these, and that's 50 centimeters across, probably. That's a cat. That's somebody holding a large Persian cat.
Right, so the waypoint 3.
see something.
New waypoint, thank you.
So the start was right on the waypoint on the two.
We needed to get an expedition.
Yeah,
But now I think we can start picking some things. See what we see.
Continuing with the transect, but we have a bit of a whoa. These fish have no fear at all. They seem to be coming really close.
ROV bridge.
Oh, yeah.
Yeah, I'll just need a few minutes. Just have to calculate some CPAs with some growlers here in that direction.
Won't be that, no.
You want to stop
Yeah, just give me 2 minutes. Stand by.
Land and have a look around then.
That one. Oh, it's different.
Yeah. A sea pen there. Sea cucumbers.
So I've completed a little bit of a transect now, and we have a bit more freedom to zoom in and explore and pick things we'd like to sample. So that sea star's probably about 20 centimetres across, which will take up a fair bit of box.
ROV bridge.
Oh, yeah, please.
Hello. Yeah, we've got a handful of growlers in the way, so we can't head directly to the waypoint. I can come laterally to starboard, and then once I clear those, I can drop back astern. So if you just want to follow me to starboard, that would work for us.
Yeah, copy that. Will.
Great, I'll start the move.
Roger.
We're just having to change one of our waypoints due to the surface ice. This is always quite a multifaceted technical achievement, moving the both vessel and ROV around, but we've got a massive extra variable here in that we're constantly keeping an eye on the ice on the surface, so we can get distracted and all excited and try and achieve things down here, but we've got to remember that there is a vessel following us on the surface, and there are some big chunks of ice around here. It's beautiful, but it makes things a little bit more complicated. So we've been a bit looser than we would usually be. Usually you plan to the very minute, but we just have to roll with the punches now. So we always have a few backup plans up our sleeve, but things are moved or cut short just as we're dodging this ice, and that's part of why it's so exciting to be in this part of the world, and that's part of why a lot of people don't get a chance to get here. It is difficult to work here, but really rewarding when it pays off. I was learning a little bit about the ice today. We've got an ice pilot on board who's a specialist in reading the ice, and also we've got a lot more experienced people here as well. And there is the sea ice, which is formed from salt water and sort of freezes on the surface, and that tends to be wind driven. And sort of moves around with the winds. And then there is the icebergs that have broken off the continent itself, and they're formed from fresh water, and they are huge. It's really hard to get a sense of scale because they're just sitting on the horizon, but usually on a vessel deck the horizon is about 14 miles away, and so these things are sitting like skyscrapers on the horizon. Apparently they're not too fussed by the wind, but they are driven by the currents, so they're a bit more predictable, but it's like buildings are moving around out here. There was one I was watching yesterday I was sure was going to flip. Oh, actually it wasn't yesterday, it was the wee hours of this morning, because it never gets dark right now either, so I'm struggling to know which day is which. And it just started to tip, and you could see where the wave action had been working on it previously, so I know one side was rising up, but I don't know if it's as spectacular as you see on the sort of documentaries. Maybe they just sort of roll a little bit and then find a new centre of gravity, but I was staring at this one for ages, hoping I'd see some spectacular skyscraper fall over and make a huge splash, but it teased me, and eventually I got hungry and had to go and have breakfast. It probably fell over immediately.
Okay, so I'll try and give you as much notice as possible.
Yeah.
Somebody else is right. Yeah. Yeah. So basically the next big rock we see rock stop. One that's probably got corals hanging off it. No, exactly.
Proper rock.
Yes, definitely. Okay, so we've got a request for scrotal blindness as well.
So at the moment we're still conducting a little bit of a transect while we also follow the vessel. We are dodging ice on the surface, and so every now and then we have to keep up with the vessel to allow them to make the manoeuvres they need to make. But we're entering the sort of sampling phase, so if we see anything exciting, then we might be able to quickly grab it as well on the way past.
We want to sample that with a scoop. If we all
So you want to pause the middle here?
yeah. There's another one there.
It's a nice little island of life. Whenever there's a dropstone, whenever there's a hard substrate really, you see a community shift, mainly to things that filter feed, things that hold themselves up into the water and catch things before they hit the ground. So on the sedimenty surfaces, you get things that snuffle and scoop up and actually eat the sediment itself. And then with a hard substrate, you get things that can hold them, like filters. They can hold filters up into the water and actually get their food that way. So all around here, we've got the things that graze on the seabed. We're seeing echinoderms, sea cucumbers, and brittle stars, and also these tube worms. But then once you've got a hard substrate, you allow these filter feeders, and I think I saw a sea pen and some crinoids, which are actually similar to starfish, but just these incredible spindly looking filter things. And they've been hanging around for ages. They were really abundant in the Devonian period. So these are some good proto-life. Amazing swimmers as well. They're surprisingly mobile. Those feather-like appendages, they sort of move them in two sets basically, and can lift off. I've got the root-like part that's holding onto the rock, and then they can move those feather-like filters and pull themselves up into the water column. There are stalked ones as well, which have this huge plant-like stem, that holds the filter apparatus higher up, and they can't escape quite so well. They're sort of committed to their spot, but having that hold fast, having that stem, means that they can get their filters way up into the water column and filter a bit more effectively. What they're all trying to do really is get their filter up above the boundary layer. So obviously water is moving, and that's really good for filtering, but then there must be a point when it stops moving, when it makes contact with the seabed. And so a lot of these animals at multiple scales are trying to get out of that dead zone, get out into the flow where they don't have to pump water themselves. They can allow it to passively flow over their filters.
Tunicate. Yeah. So, there's a few things we can collect here. Yeah, position. Yeah, position where you can get access to the rock.
One of the sea cucumbers has decided to check out. So they can, well, some species can swim. And there's this undulation of their mouth and appendages. They sort of splay it out to, it looks almost like climbing through the water rather than swimming. I'm not sure if we caught it on the video there, but what they tend to start with is dropping ballast. So the first thing they do when they decide to take off is usually a massive poop.
Well, that's a tunicate. We won't collect it because it's full of pieces. It's a nice looking one. Sponge or something. ¿Hay anfipod?
¿Ah?
¿Tuviste el anfipod?
Sí, pero la piedra.
Mira, eso, an ice pole there. If we could get suction in there, that'd be good. That thing. Suction. And if you get the off-road as well, it wouldn't be the end of the world. Yeah, okay. Okay, fine. We'll stick it in section 6. Or 5. Yeah.
5 or 6.
5 or 6. We don't need a 5, right?
the isopod's the target, but anything else with it is fine.
So it's next to.
So it's there. Uh-huh.
Yeah, okay.
Yeah, okay. And get this isopod first. Right at the limit. It's here. It'll be just a few centimetres in front of where you are. There you go. Yeah, he's gone. Yep.
You got it.
Right. The next one we spotted is here. And it can go in the same section as that. Yeah, this it's amphipod on top of that. It's like a browser. Oh, there's two of them. Yeah. There's two in there. Yeah. Yeah. Back a bit. Yeah. Right, got a good grip there. There he goes. If he comes out, but yeah. Let's see where his mate's gone. There you go. Yes. Exit.
It'd be interesting if they know.
Which yard it was?
Huh?
Which yard?
SS6. Let's have another look around, see what's. Keep it in your grip. I mean, let's have a look at the top surface of the bigger rock. Let's see what's up there. Yeah, that's an urchin. Yeah, that's an urchin. Looks like another amphipod there. Yeah, but we've got two of those, that's enough. Crinoid, another amphipod. Did you suck the crinoids? Do they suck up? I could do with the crinoid. And then they've got polychaetes there. Medical, yeah. Two polychaetos, ¿cierto? Esos son serpulates, yeah. Serpulate. It was 5.
5. Single. 5.
Yeah. Crinoid and suck?
And a 6.
Yeah.
Okay.
Yeah. Let's go. Yeah. Give him a nudge. There we go.
Bring it to you. Tunicate. There you go.
There's the granoid. Excellent. Did you get the amphipod as well? Yeah.
6?
Yeah. And we want the polychaete worms, the tubes that are there, but I think they're probably going to biobox. But what do you reckon the best way to get those are? These tube worms. I'm not entirely sure, but I don't think they'll suck because they're probably cemented to the rock. I think we're going to have to scrape them off somewhere.
Okay.
Yeah. I mean, maybe the scoop. That would work.
Pour it into the
And then tip it into a biobox.
box or the
No, a biobox, I think, for that, yeah.
Yeah, stow it.
Scoops in the rock basket.
lo están mostrando con la aspiradora,
No, vamos a usar el scoop para sacar un poquito de scoop.
Cristian quiere un chanchito.
Sí, yo sé. Sí.
Ese chanchito que está ahí al...
Los crotoplanos.
Sí.
Yeah. But polychaete primero.
Porque es chiquitito, no tan grande.
Scoop. Yes, yeah. This area of polychaetes and various other things. I'm gonna stick it in biobox.
Are we using the metal one?
No, the scoop.
Scoop it in the rock basket.
Scoops in the rock.
Yeah.
Right here.
Because they're kind of stuck to the rock probably, so just probably have to scrape them off.
Yeah.
Yeah. I'm gonna stick them in biobox 4.
Probably from the ground itself.
start outside and run it along the rock.
Hold on one second. I'm just trying to reposition. So that's the stuff that needs to be put.
Yeah, this stuff here.
That's what I was gonna say. I think you probably either want to grab like right there. There with the pocket, coming down. I can hold it together. Whatever. Whatever it is here for you.
What did I see with this? It's not good at that one.
Sure. It's gonna slip. You want to get that vertical shaft in the pocket. Right there, there you go. Nice, perfect. Now you can straight.
When I got rid of this.
Yeah.
What's the corner is not that way.
Yeah. On the way.
Excellent. Yeah. So firebox for something.
Clean.
Yeah. So,
Watch out.
One of this move, 7 points.
Yeah. What'd you say, 4A?
for a, I mean they're all empty, so whichever is easiest to drop it in.
Place a corner to
Yes,
I mean, whichever one you want, and we'll make a note.
I'm racked in on the way too. Don't see it, man. Probably has form. Metal form. There you go. There you go. Just like that. Yeah, I know, right?
There we go. There we go. Excellent.
Well, you're not gonna put that in?
That's the guy.
I'll get you.
Well, we want one of the sea pig, but I think that's out of reach, isn't it? Certainly from that arm. This one here. But
Well, we just hop over. I mean, if you want to.
yeah, I mean these are relatively.
Yeah.
Well, no. In this case, it's not so fussy. Yeah, it's for our collection, not for Teresa.
No.
Yeah. But I mean, it's not biggie if you can't. It's just they're quite common and get one later. It's right here on this side.
Try to grab it with them.
Swap to the other arm. There's some coral in this rock.
It's second in it. It's like
I think it might be what we got earlier.
cucumber.
Yeah, we looked when I was there, but I'm not the expert.
I figure that's the transecting going.
Fine, we did the full transect from between waypoint 1 and waypoint 2. Just a few bits and pieces.
Okay.
Just get that sea piggy and then here.
Yeah.
That guy there.
Yeah.
Yeah, that one. Yeah, again, stick in buy box 4 when you get it. Four B's fine.
B. 4B.
4B. Thank you very much.
Alrighty.
See you later.
So we got these over here. I'm using them, just trying the new views for sampling, but we got the pads and put it over here. It's got two Roger starboard. So this is under the date.
Thank you. Thank you, America. Have a good break. You got it.
And my back.
I have to go get a.
It's okay.
So what we asked is
Okay, I need to stay here.
so we did this transect and then I can start there, you have 3. Put in the waypoint for the ship, and they report back that I can't quite go, take you there.
The queen is, who is this one?
So
Pero la princesa, ¿o quiere ser la reina?
transect 3, the ship may have to move just gotta follow along and record. So you can see their trail. Wasn't, that's where we stopped after 1 or 2. This is the direction that we're heading.
I'm back. I guess you can just head along the waypoint track to the next waypoint while I'm getting organized. I'll put these on. Hi everyone, my name is Erin Easton. I'm from the University of Texas, Rio Grande Valley, and I will be your watch lead for the next 3 hours or so. We are on our first station, dive 777. We have an estimated recovery time of 2 a.m. I believe. So this is our third dive of the Climate Connections at the Icy Interface Cruise. So we're off the coast of Antarctica and the Bellingshausen Sea. We started, I think, around 700 meters depth, and we're going to work up to a local shallow point of around 500 meters. We're surrounded right now by a number of icebergs. And just trying to see what we see here on the seafloor in this area that hasn't been explored yet. At some point, we'll be collecting a lot of push cores. Yeah, we can keep going towards waypoint 2. Thanks. Sorry. I need to get my one ear out of the headphones.
Ruby Bridge.
Waypoint 3, my bad.
Waypoint 3, but I'm sure maybe some ice between us and there.
Yes, that's correct. I'm going to have to just come to starboard, so move laterally to starboard and then come astern rather than make a straight path there. That's right.
That will do.
I just
Great,
gotta
so
check where the icebergs are.
Follow
so
me
we
around,
can
that'd be
see.
appreciated. Once we get clear of these few growlers, then we should be okay to move astern.
Roger that. Just be clear, you're going to move it straight north just about.
Yeah, exactly.
You got it, Luke. Thank you very much.
Thank you.
So we're just gonna follow them for a while. Just let them know when we can go back to the line.
Cool. Yeah. Yeah, just going to go. I think he said straight north, and then we'll have to stern over. So push northish for.
Okay, sounds good. Yeah, so we're just having to modify our track right now because there's an iceberg along our track, so we're just gonna meander around it. Yeah, today's dive, our goals are to get another set of 17 push cores for our biologists who work with meiofauna and macrofauna on board, and our geologists who are on board. And then hopefully we'll find some coral species we didn't collect on dive 2. We might also be collecting some holothurians for one of the biologists on board, some water samples as we go for eDNA. We just did a shift change. This group will be with you for the next 3 hours. We're still waiting for one person to come in, but I think they're processing their samples right now. So right now we have myself, Anna, and Maritza. So I don't know if anyone wants to say hello. That was Anna.
Hola a todos, aquí otra vez Maritza Castro, bióloga marina. Esta es mi 5ª vez a bordo, así que espero que disfruten este dive.
Maritza, what is it? Do you think so? It might be. It's a big one if it is. Yeah, it looks really unusual. Yeah, if we can zoom in on that really quick, because that's a coral of some sort. Well, yeah, it is. Yeah, it's this type of mushroom coral here. Yeah, this one's different, so let's go ahead and scoop this one up and put it, if we can.
Are we bridge?
We are pausing for a second.
No worries, I'll stop.
Yeah, this one I haven't personally seen before, so not 100% sure what it is. I'm not sure if it's known for the area.
Do you want to put this?
How big is it with the lasers on?
But it's soft, so.
It's soft, so yeah, if we can grab it from below and pop it in a coral quiver, that'd be great.
I won't crush it if I pick it up.
I don't think so. These are those ones that, like the mushroom, you usually grab the base of the stalk and tear it off a rock when it's on a rock.
Okay.
So I think if you get below the mushroom head.
Can you put it from one of these four?
Yeah, you can put in one of those four. And your coral quiver of your choice.
Okay.
Mm.
One for child?
Yeah, so it does look like it's something like Anthomastus. There's actually another group, I think, that's found in Antarctica that's similar to Anthomastus, that it might be instead. Fine, I was just looking at the group yesterday.
Oh, so you want to put it on the rail?
Yeah, put it on the rail.
I'm gonna put it on the rail.
Okay. Sounds good.
I just stood this.
I'm gonna shift over to the right.
Yeah, sounds good. Yeah, man, it's coming forward a little bit too.
Alright, yeah, so we're just getting repositioned here to take this sample. Yeah, we have a number of dives that we're planning to do off the coast of Antarctica here, I think. We have a couple weeks where we're doing dives on stations. We had quite a long transit out here. Let's see, I think we left on the evening of the 9th and we arrived... What's today's date? It is the 19th. So I think we arrived on the 17th. So yeah, we had about 8.5 days of transit. It was quite lengthy. We had to go the long way around out from Punta Arenas, Chile. So we had to go to the east and then come down because we had some bad weather to avoid. And yeah, I think if this isn't Anthomastus, there is another genus that's very similar to Anthomastus that's reported for Antarctica. I just can't remember what the genus is, it's like hetero something. See if I can find it. Yeah, there's another genus called Heteropolypus that's known from Antarctica. And I would have to look up the differences to know for sure which one it is. This one looks different than what I'm used to seeing, but for Anthomastus in the Southeast Pacific. So I'm sure it's a different species than what we're seeing up there, but yeah, I think if you just grab, that looks good. It looks like you're going to grab below the base of it, or below the polyps will retract into that top mushroom part. Uh-oh. Oh man, it's stuck. It's on something, attached to something strong. Oh, there it comes. There it goes. There we go.
Do you want to give a good video?
Yeah, a glamour shot, please. Thank you. Yeah, that's lovely. See the polyps there.
Happy?
Yeah, that's great. Thank you. Appreciate it.
Okay, let me reload some more.
Yeah. Yeah, coral.
I explained to her.
Savetta just joined us, geologist or geochemist. Yeah. Let's see, UTC time. Where is my hat? Thank you. UTC depth 666 meters. Where's UTC time display? There it is. Thank you. 16. Alright, so I have to put UTC. I want UTC time and depth, right?
Yeah.
That's it? Okay. Alright, so other things on our watch list tonight are mollusks and polychaetes. Looking for different corals we haven't seen yet. We're not like under the ice. There are icebergs in the area, and not that long ago, before this part of summer, this area was under ice. We have really high productivity here. You can kind of see the greenish on top of the sediment, and so that's really productive right now. There's a lot of phytoplankton and it's raining down. Alright, thank you. We can go ahead and go back to our transecting position for the camera with lasers on and proceed. Thank you so much.
Lasers on.
Yep.
Okay.
But like right now we're navigating around an iceberg. There's a low-lying growler not too far off the ship, so we have to stay clear of those.
We will make adjustments as they make adjustments.
Yeah. We're just gonna go basically straight north.
No, not straight north.
Yeah, that's what they want to do.
Okay.
Yeah.
I'm just watching your position.
I'm seeing a number of really silvery fish here. I'm sorry, I have no clue what they are. Our resident fish expert did a deployment of cameras last night, or lighted camera system.
On the move.
I think he collected some 40 or so hours of video.
All right,
Seeing
I
some
will
anemones
continue to
here.
come north and slightly east, northeast-ish.
That's what we're doing.
You got it, northeast.
Number of holothurians. Looks like we have a nice sponge there on the left. Is that like a fish playing? What is that? Interesting. We collected some of these primnoid corals earlier today. I think we collected at least 3 species. There's definitely 2 distinct species. The third was a little hard for me to tell right now. Also managed to get, I think, the full complement of 20 push cores on our last dive. So
Hey, look,
scientists overall were quite happy.
Waypoint 3, and if you're gonna make any trajectory, just let us know, and we'll just follow accordingly. Okay.
I know he wants to go.
Yeah, you'll probably have to come north of that line because I'm going to head more north than northeast.
Roger that.
Thank you.
Roger that.
I also collected a number of holothurians earlier today, including Solus, which is a type of holothurian that attaches to the rock. That is, seems to be a slight shift in the benthic community right here. Some crinoids. Also been seeing a number of them. So yeah, this is a 36 day expedition. We ended up leaving port a little bit early, so we try and get ahead of the weather. So we left the night before we were originally planned to leave, which is good because it took us a little longer to transit. Savannah, do you want to introduce yourself and tell them a little bit about what you do on board?
Okay.
Alright, so I'll let Savannah tell you a little bit about what she's doing on board.
Hi, so I'm here as a part of the geology team. I work in the University of Cambridge and also at the British Antarctic Survey. And here I'm working with sediments, and I'm specifically interested, I'm a foraminifera geochemist, so I'm interested in these little microscopic shells and particularly studying their chemistry. And we'll also be doing some pore water experiments, so extracting pore waters out of the sediments and comparing their chemistry to the little shells in the sediments. And this will be to study past ocean climate changes, especially to do with oceans and ice sheets on Antarctica, which is very useful to test the computer simulations, so we can use past climate geological stuff as a natural experiment. Yes.
Awesome, thanks. And that's what you're doing with the push cores that you're collecting, or the gravity cores?
Yes, so all of it will be collecting some so I'll be taking the sediment back whole, apart from the pore waters that I'm collecting out of them, and then I will be processing them back in Cambridge together with other collaborators and some students to get essentially these forams, or get rid of the sediments and find my little shells. And then eventually I'll analyze them using chemistry instruments such as mass spectrometers for various chemical parameters, and yeah, reconstruct past climate changes in this particular area, which is very interesting because we don't really have any data at the moment from the Bellingshausen Sea.
Thank you.
Just heading slowly north of the line. It's exactly
Okay. Yeah, that's fine. We'll find our way back over in that direction once he's good. Yeah, there's quite the. Ooh, we have a. Can we zoom on the sea pen here? Umbalula.
Yeah.
Seabed.
Yep. Nice. Good. All right, we're good. Thank you. So, Sveta, how long does it take you to process a sample?
Depends on what type of sample.
Okay.
But generally it's quite a slow process because we've got to sieve these samples first. So this is kind of similar to a child in a sandpit. So we just sieve them to get rid of the finer mud and to, again, remain with the sand, so the bigger things and these shells that I'm after. Then I've got to find the correct type of shell because there's loads of different species under the microscope. To do this I use a really fine paintbrush, so that normally just has a few hairs. After that I've got to chemically process these shells, which can also take like a few weeks. And then eventually we get to the measurements. So all in all, it's kind of like, if it's a short push core, so around 25 centimeters of sediment, can be a few months of work just to get data out of that one core. And even then you don't know obviously what the data shows until you actually obtain the data. Because geology is a very kind of observational science, so you have a hypothesis, but essentially we make observations of the data we collect, and then sometimes it's radically different to the hypothesis.
All right, thanks. That was kind of cool. There was like there's some like mostly buried sea stars here. Sure, let's go ahead and zoom in on one of them, and then maybe on one of these urchins too while we're at a pause. Yep. That's right, while we're waiting for it to clear, maybe we can zoom at this urchin over here. Just to get some information. Might help with any IDs when they're working on the annotations. I think, well, I thought it was an urchin, but now I don't know.
It's urchin. It's a
Yeah.
regular urchin.
Yeah, an irregular urchin, right? That's what it looks like to me. Kind of looks like a taco.
Yeah.
With some hair on it.
Copy that.
It is interesting. Cool. Yeah. No, we're still waiting for the other part to clear. What else can we zoom in on and look at really quick? There's some ophiuroids laying around. Small ones. Whatever. Is that? Oh yeah, that's like a crinoid, huh? Yeah, look at them, well camouflaged. Yeah, our cruises. We've had some beautiful weather on the cruise. It was a little windy yesterday and cold. Sure, y'all.
Starfish?
Yeah, the starfish cleared.
It's got a nice fine layer of air on
Alright.
It's a mystery, I think.
Oh, okay, if we see. Okay. Yeah, here, just let me know. Yeah, if you see him, you can just say it out loud.
Oh,
You don't have to type it.
right.
That's fine. I don't mind.
I was just telling Ellen I'm really interested in some brachiopods, so they're bigger shells, so that chemistry.
That looks like a track.
Yeah. I don't know. It's kind of cool. Yeah, maybe. Well, it's cool. Yep, it's waving at us. Yep, I'm good, thanks. Let's go ahead and continue on the transect and see what we see.
Exploration.
Yeah, there was a worm tube over there too, yeah. I didn't see anyone hanging out.
On the move.
At least two, I think at least two different species, maybe a sea cucumber here. I think these might be the type of sea cucumber that were collected. No, I think they were collected earlier. Yeah, I can do a quick zoom. Oh, there's a fish there too. Let's do that. What's that? Cute fish. That one almost looks like, oh, okay, I see. That's his dorsal fin. I was like, it looks like a skeleton kind of, but it was his dorsal fin. Yep, we're good. Thanks. All right, we'll go back to transecting. Did Tom make it? Oh.
Hey, I think that's oceanographic. I've got a little lure on the top lip.
Okay.
The bottom lip, sorry.
Yeah, you want to step up to a microphone? There's one over there. You can tell them all about what you do, Tom. So yeah, we've got Tom here, our fish expert, so he can kind of shed some light on some of these fish we've been seeing. Carlos will get you set up there.
Hi everyone, I'm Tom Lindley. I'm a, fish guy, I suppose is the easiest way of saying it. I'm a fish curator at the Museum of New Zealand, Te Papa Tongarawera, and we just caught a little sight of a Pogonophrine, so one of these little mottled fellas that was sitting on the seabed. And the cool thing about those is that they have a lure, like an anglerfish. They have something they waggle around to try and entice things close enough to their mouth, so they lay very still on the bottom and then wiggle around this lure. But unlike the anglerfish, where it comes out the top of their head, it's actually a part of their bottom lip. And there's lots of different shapes, and they're kind of a bit raggedy looking. Usually the lures on an anglerfish is quite specific. It's sort of pretending to be a squid or pretending to be a copepod, but these tend to be like a little pom-pom, and part of the reason for that is they accidentally eat them sometimes. So when they snap at their food, they sometimes get their lure as well. So sometimes they're missing, and sometimes they're showing a little bit of damage.
The same one as before. So amazing. Yeah, we're good, thanks. What about some of these silver ones we've been seeing? There's an abundance of them.
Yeah, the silver ones don't seem to show any fear, actually. They've been a bit of a nuisance. So they are the Antarctic Jacobfish. I've got the Latin name for you, for all your Latin heads out there. Notolepis coatzi. I'm not sure how to say that second bit. Anyway, those are the silvery guys that are constantly buzzing in front of us. When they're startled, they quite often face plant into the seabed. I think they're probably disorientated by the lights. This is probably more light than they are ever exposed to. But when they're more relaxed, they are swimming. Well, they're holding their body quite rigid and then just undulating the very last part, just the tail. So really interesting swimming gait. And we see that in a few deep sea fish, and there's a few different reasons why they might do that. One of the ones that we're thinking about is actually that it is to allow their lateral line, allow those sensory cells along the side of a fish to pick up vibration without interfering it with their own body's vibration, because their brain is constantly having to cancel that out. They have to cancel out their own movement in order to detect vibrations in the water. So this big long body has this huge sensor on it essentially, and if they just move the very tip of their tail, they get a nice clear signal down that body length of any vibrations in the water.
So I think, I'm wondering if these are some of the... Thank you, Tom. Appreciate it telling us about those. Me either. So the Arctic Jacobfish.
Yeah.
Okay. I'm sure I remember it. Is this a touch away anemone?
Yes, it's an anemone.
Yeah.
Looks like a Liponema, but I don't know.
Yeah, I don't know either. I think this is an anemone at all tucked in. And get a nice little zoom and see if it can be used for identification purposes later, but I've been seeing a number of these. Moritz is going to do some research for me. All right. Thank you. Hopefully that's a few seconds that they can also use for the highlights. Yeah, we can go ahead and proceed. And then we'll just transect here a little bit. Keep on transecting until we see something interesting. Not that most of what we're seeing isn't interesting, it isn't actually, but maybe something new, I guess, new and interesting that we haven't seen yet on the dive. So yeah, we have quite a large group of scientists. I think there's 24 of us on this expedition, and a quite diverse group from oceanographers and geologists. So we have people who are doing mapping, and we have people, let's see, we got mapping, oceanographers, like deploying some gliders. So at some point, hopefully, those doing that work will pop into the room and can tell you a little bit about what they're doing. We deployed a...
Lara is actually here because it's deployed the gliders, so you can get her to...
Oh, Laura in here. See, don't have eyes in the back of my head. Hey, Laura, you want to tell us all about your gliders and what you guys did the other day with the... Let's catch up our audience on what we've been doing on this expedition. Hola, Maxi. Yeah, should be good? Yep, she's good.
Yeah, hi everyone. I can tell you a bit more about the gliders, or, well, I guess they're a type of autonomous floats which literally glide through the water. We can pilot them from land. We actually have two teams piloted two different types of gliders. Right now only one is in the water, and they are from the British Antarctic Survey and the University of East Anglia, both in the UK. I'm based at the University of Cambridge and collaborate a lot with both institutions. The glider that's currently in the water is a sea glider. The boarding name is SG558, sorry, but yeah, apparently the boarding name, we call her Bella, which is a bit easier also to remember. If you're curious actually about her whereabouts in Antarctica, you can actually look her up on the University of East Anglia website. Look for Sea Glider UEA and it should pop up quite easily. And you can find the mission where we deployed her. We deployed her a couple of days ago. She's doing fine. She has already completed 20 dives. So the glider works with a buoyancy engine. So it basically pumps oil and moves a bit the battery to change the internal volume, and so it changes also its density, and so it can become buoyant or become denser, and so it starts like zigzagging basically through the water, and every time it's at the surface it transmits the data that it has collected, and at the same time it listens to what we have to tell her. So she can receive information about what to measure, how frequently to measure things, and also her new waypoints, so we can change her trajectory. And in terms of what she actually measures is a number of sensors. She has a number of sensors on, including temperature, salinity, oxygen, fluorometer, and backscatter, and PAR. And this is all to measure the physics and some of the biogeochemistry property of the water. The goal our goal, let's say, as oceanographers in these expeditions is mostly to monitor the glacial meltwater that is actually coming off the shelves because this is an area where ice shelves have been retreating rapidly, and so that leads to a lot of glacial meltwater, and we want to basically better quantify how much is produced, where does it go, where does it flow, how is it changing also the biogeochemistry in the area. And possibly beyond, because this meltwater apparently then keeps flowing around Antarctica and can have also global effects and not just local ones.
Thanks, Laura. Awesome. You can keep talking if you want.
I can keep going if you want.
That's really fine. You can tell them more.
Well, more of the things that we're doing. Yeah, I mean, we have another glider that is going in the water in a few days. That's a Slocum glider from the British Antarctic Survey. So that's not in the water quite yet. Actually, that will stay in the water for much longer, and it will be deployed in a week time, more or less, and it will stay in the water until June, more or less. So in general, these gliders are quite nice because then once they have been deployed, then can stay in the water for months. When again we're flying autonomously, so we don't necessarily need to follow them. Bella, for example, will be in the water now for a week time, more or less, until we move around this part of Antarctica, the Ronne Entrance. And we will pick her up in about 7 or 8 days and then deploy her again. We also have floats. These are different types of autonomous gas vehicle. They will also give us profiles of temperature and salinity and kind of fix the location wherever we deploy them. The nice thing is that their battery have very long endurance, and so they can stay in the water for years. So we will monitor basically this area effects with these floats for 2 to 3 years at least. And when we sample through the CTD with conductivity, temperature, and depth, which is another technique where we measure both, again, physics and chemical properties.
Awesome. Right. Yeah. Again, we're collecting this polychaete here right now on this sponge. That does look like it has at some point fallen over, and also has maybe a tunicate on top of it, some hydrozoans.
Okay.
Show
me which thing this was.
So, I'm sorry if I missed it. I was also listening to them, Laura, but the other glider that you're deploying, right, gets to be recovered by the base here, right?
Right. The other one will not be recovered by us. It will stay in the water, and then it will be picked up at Rotorua, which is one of the stations in Antarctica led by the British Antarctic Survey. Yeah, which is exciting. It's going to be in the water for 4 months, more or less, at least.
Yeah. So do we know which station you're planning on deploying that one at?
Probably station 6. So right after we will leave the Ronne Entrance, and basically, it all depends on sea ice. As also for the ROV missions, we see it moves quite a lot and can move rapidly. So if that is going to be open, it will be deployed there. So in about, I guess 10 days or so, it will be in the water.
Okay. And then we're recovering the other one at station 9, which is like maybe 5-7 days away.
Yes. But we hope to deploy Bella again for a shorter deployments once we are around station 6. So station 6 and station 5. So we'll do multiple deployments with Bella, but short ones, and then one long deployment with the Slocum glider.
Okay, that's cool.
We're still looking for names for the Slocum glider. So if anyone has suggestions, also.
Let's see. Oh, sorry, Matt. It might be escaping us. I tried, Matt, to get you your polychaete. No.
I'm going to shut off and I need to turn it over. Flip the sponge? Flip the sponge?
Yeah, you can try. I don't want to take too long, but you can give it one flip and see if we can get it.
So you can try to pull up a little bit. You see that pull up?
Push it off.
I think the polychaete or something escaped. Yeah, I saw this escape. It might. I'm not sure, to be honest. I thought maybe it could be. We're just righting the world. You see him? Yep, there it is.
Yes.
Yes, got him. Yes. All right. Yeah, perfect. Thank you.
Next to 1. Nothing's more than 3.
All right, perfect. And we are at 55 meters. success. No. And when you have that stored away, could you grab a Niskin bottle? Let me see. So yeah, as you can see, we have quite the diverse team on board. And like I said, we have a lot, I think we have 4 teams that want push cores to do different things with them. So biogeochemistry, geology, meiofauna, macrofauna. We have a few people on board who are working with the larger fauna, so myself working with the corals. We're focusing primarily on octocorals this time. Just because that's my main focus, and I don't know if I have someone to work on all the others, so we're doing that, maybe a little bit of block coral collections, other corals of interest. We have someone working with holothurians on board, and then we have a couple who are working on just general collections for species for museums in Chile. But yeah, I think we can call this like mission accomplished, you know, sponge uprighted.
Yeah.
It's like the leaning tower of sponge. Oh my god.
See, it's left in its natural state.
Yeah, we're just preparing things as we go.
Yeah, that's it. Saving the sponge.
Our friends, the sponge.
Look at the sponge. It's so happy now.
Yep. Okay.
And Anna wants Niskin.
Yes. Thank you.
So yeah, Anna's getting a Niskin bottle collection here. I don't know, Anna, do you want to talk a little bit about what you're doing?
Yes.
And of course, anyone, you can say it both in English and in your native language if you want, of course.
Oh, il buono in Italian. Ok. Ciao a tutti, sono Anna Maria Damo, lavoro alla Northern University in Norvegia e in questa spedizione oceanografica stiamo lavorando con, appunto, come diceva Erin, con diversi colleghi, con diverse molto multidisciplinari, perché abbiamo geologi, oceanografo e biologi a bordo. Io personalmente sono una biologa marina, ecologa marina, e l'obiettivo principale in questa spedizione è quella di analizzare l'acqua del fondo marino e per due obiettivi principali. Uno è il DNA ambientale, quindi cercare di caratterizzare l'habitat di questi fondali marini che in effetti sono poco studiati ed è difficile raccogliere e campionare tutti gli organismi che possono caratterizzare l'habitat, quindi l'idea è quella di appunto di analizzare
You guys were number 6, correct?
Scusate un attimo.
I kept hearing the word 6. Yeah, no.
Uno, yeah. E una parte è appunto analizzare e caratterizzare l'habitat, e in questo modo l'idea è quella di usare il DNA ambientale che si trova appunto nell'acqua dei fondali marini. Inoltre la stessa acqua verrà anche utilizzata per campionare microplastica. Come vedete ci sono spugni, altri organismi che comunque si nutrono dei organismi che sono nella colonna d'acqua, e quindi l'idea è quella di vedere se la microplastica è anche presente nell'acqua proprio sui fondali marini. È stato trovato in vari lavori che la microplastica è presente nei sedimenti, quindi l'idea è quella di valutare se anche qui ci siano delle microplastiche che possono effettivamente dare effetti negativi all'alimentazione di alcuni organismi. E niente, tutto qua. Altro interesse sono anche in particolare i coralli e tutti quegli habitat biogenici che ovvero che sono costruiti da organismi e quindi ovviamente in questi fondali così nell'Antartide, dove appunto l'acqua è molto fredda e non ci si aspetta magari di trovare tanta diversità o tanti coralli, in realtà anche qui può sorprenderci trovando nuove specie, ed è quello che effettivamente probabilmente abbiamo trovato oggi raccogliendo appunto 2 o 3 campioni di coralli che sembrano essere almeno 2 specie diverse. Però questo lo sapremo dopo con l'analisi di Erin Easton che farà più avanti. Over to you, Erin.
Bridge RV, just on the move again.
Yeah, sorry for the cross-talking a little bit there. Just you couldn't see it, I don't think, in the main camera, but we saw a squid in the other camera, and it swam off before we could get a good look at it. Thank you, Anna. So yeah, we're just going to do a little transecting here for a little bit. We've been sampling for a while. We want to kind of stay on track. So we're going to work our way towards waypoint 3. Our goal is to get in the area of waypoint 4 to waypoint 7 today, where it's a little bit shallower. And there's some interesting like scar marks from the ice having moved, like deep ice moving past the area. I'm not a geologist, so I'm sure they can explain it way better. I don't know if Sveta knows anything about the scar marks that we've seen, or is that a question for someone else?
No, I'm afraid
Okay.
I do not know about this, sorry.
All right, well, maybe someone else will shed some light on it later. So yeah, we're still seeing a lot of brittle stars, lots of animal tracks, lots of evidence of the high productivity in the area with this greenish coating to the sediment. So you can see where the animals are kind of clearing that all out. Like the worms there, you can see little circles where it's lighter in color. And it's settling in some of the depressions and older tracks as well. And so Maritza is, I don't know, since you were last on the ship, you graduated? ¿Cuándo terminaste su título?
En agosto.
En agosto. Ah, okay. Antes que el último viaje.
Sí.
Yeah, so shortly after our last expedition in August, Maritza graduated. So she's an official marine biologist now. So she was able to come and help Anna and I out on this expedition. So that was amazing. We found out she could join us just a few days before we set sail. So she immediately changed her plans, packed her bag, bought the required equipment, and like 4 days later, met us to get on the ship. Another interesting story there. Pretty sea cucumbers.
Web tubes, right?
I'm not sure what species is in that. Like, I really don't know.
I would say they're like carbonate.
Yeah, like older skeletons. There's another sea pen or umbrella.
Hey Luke, what's a good distance for you to continue?
Oh, there's a little fish all curled up.
So that's why I'm not sure if I'm not
Probably not going to be able to get right on waypoint 3. Probably going to have to be north of it.
A few more
Yeah,
corals are
basically,
starting to
I'm
pop
good
up
where
again.
the ship is now, or we're going to have to move several ship lengths to the north. We've got one big iceberg and several small growlers. So yeah, probably not going to be able to achieve waypoint 3.
Okay.
it's all good. We're just trying to figure out what's going to be the best lines for us to run down there. So in terms of us making progress on this line, ultimately it's going straight north going to be the best route.
I think so. It's hard to tell because all the icebergs are moving in different directions at the moment too. But yeah, probably north is going to be the safest bet.
Okay.
We need 100 more meters to make it to waypoint 3. We're on a heading right now of 53 degrees. So you think we can make this, or do you want us to head directly north?
Just figuring out how we have to go.
Budge closer, and then
Nice
we're able to
yellow
get there.
sponge, I think.
We might not be able to stay for too long, but I'll keep you posted.
Okay. So I'm gonna keep my heading, okay? 5:30.
Yeah.
So yeah, for a little bit there we were seeing some corals again. I'm not sure if they were the same as earlier, but it wasn't a good moment to stop because of the ice. They're trying to figure out how we get around the ice right now. So we can't quite make it to our next waypoint because there's an iceberg in the way. So we're going to modify our path a little bit here. Here's again.
Hey Aaron, is there a feature on waypoint 3 or is this just a transect line?
I think it's just a transect line. If we have to go around it, we have to go around it. Alright, we would just want to make it up to that, like the green area. So I think if we're off one way or the other, it should be okay. There's just some scars across the area.
This guy right here.
Yeah, this is just like an interest between, once we get to 4 is where it's more interesting. So as long as we make it to 4 somehow. Yeah.
There it is.
Ooh, there's a squid.
Yeah, I didn't know what it was. Long darted itself into the sediment.
It's like squid darts.
You want squid?
I know. I don't have any way to preserve a squid. And Fernando's not on board. That was pretty impressive. Yeah, we did. We had a squid cephalopod expert on one of our expeditions, so we did catch some squid.
We caught some big squid up on northwest. Ordinary giant squid.
Oh, nice.
Yeah, almost.
Crinoid.
So if we don't need to go to 3, I might check with Luke to see if there's the best path to get to 4, if that's okay. Already right.
Yeah.
Okay.
Yeah, if we need to go north or south of waypoint 3. We can sit here if you want while we wait for them and look at this coral. Or I can find something else shortly ahead to look at if it's too late.
It's never too late. Alright.
I'm just gonna sit here for a second.
Hello again, Luke. Week 7, contact with the science team, and the priority is to get to waypoint 4. Going to waypoint 3 is not a necessity. So whatever moves we make probably should be bent towards that priority if that gives you any more information. What do you want to see?
Okay, cool. Yeah,
That
let me
core
just
right there.
take some CPAs, closest point of approaches with a few of these icebergs around it. I'll let you know. For now, we're okay on this heading, or this course.
Roger that, thanks, sir.
Thanks, Tyler.
Is that a... Actually, can you go back a smidge more? Because I'm beginning to wonder if... Yeah, I know it's not super helpful, but... I wanna... Right here, that pink.
Okay, alright.
Oh. Oh, okay, yeah, it's just one of the worms tucked in. Okay. Yeah, we're good.
Okay, so
Yeah.
you want to look at the other one?
Yeah, let's go and look at the coral. It just looked different. I thought it was something else, but... While he's calculating the course for us. So yeah, this might be one that we collected earlier. It's got multiple branches, but it's a little hard to tell.
Or is that brachiopods?
There are some at the base of there.
Think we can try and collect me a couple of brachiopods if possible?
Yeah.
You want to pick up the rocks?
Let's pick up the rock and get everything that comes with it.
Okay.
And we can put it in
Bridge, alright.
a quad.
Copy that, I'll stop.
Which one do you want to use?
I guess about 4 arm right there.
Yeah, and just do a biology observation with the coral and brachiopods for the collection and whatever else for the sample.
Okay.
And then it'll be plus rock. And then others should be on board, should be happy getting the bonus samples for the museums. I think this is a different crinoid than we've collected earlier today. There's an oaf on there. I'm honestly not sure it's the same species we collected earlier of Primnoid or Thorella. It just looks similar. It's got a couple of brachiopods at the base. 3, I think. I think you're getting 3 for 1 here, Sveta. There's a 2.
Would be brachiopods. Wow. Let's get excited for some fun chemistry.
Look at that.
You're
nice.
Okay, we're gonna give them a good Photoshop.
Yes.
Happy?
Oh,
nice.
Smile. For Anna. Happy, happy? Okay.
Yeah, can we zoom in a little bit on the coral in the background also? Thanks. There we go.
Oh, look at the pole.
Nice. That's a coral glamour shot right there. Can we zoom up just a smidge, see what's on the tip of that?
Yeah, it's the same that we took this morning, right? You had the similar
Nice, thank you.
at the top?
Yeah.
Now we're good.
Watching it.
So we got C and D are still.
Yes, whichever one, your choice.
Your choice.
Yeah, I wish too. I think the thing for me is I want to properly anesthetize and euthanize the squid. So that's even worse than having some way to preserve them properly. It's having someone on board who can properly deal with the specimens.
No. When now.
Looks good.
Yeah, no.
You're sure. B, for B. Brachiopods, coral,
That you can entertain on
Yeah.
for a little while.
Okay, awesome. Thank you very much. Alright. So what's the word on the direction we get to head? For?
We said this heading's fine for the time being, so
We're
Okay.
going for waypoint 3.
Alright, sounds good.
whatever.
Keep the
Yeah,
current
looks like-
heading.
Yeah. We'll just
Looks
kinda
like.
check in as conditions change, I think.
Okay. Perfect. So we're gonna go ahead and
We're heading for waypoint 3.
keep heading towards our next waypoint. In the time it took us to do some sampling and everything, it looks like the way is largely cleared to waypoint 3. And we'll just keep checking in as we move towards waypoint 4.
Bridge, RV, we're moving. We're going to continue on in this heading until you say otherwise.
Hey Tyler, yeah, sounds good. A couple growlers have passed the stern, so yeah, we'll just continue on this track, and then if I have to deviate, I'll let you know.
Your friend's big eyelid, thank you.
Thank you, Tyler.
So yeah, the whole time I think you'll find like we don't have really set times on when our dive times are going to be a lot of times because we have to deal with the ice conditions and navigation from point to point. So for those of you who are used to tuning in, you'll find that our estimates of when we're going to dive again might not be so accurate. You just have to kind of like hang on and wait till you see we're going live. There's just a lot more complexity. We plan to go in a dive location and then can you do a quick zoom here? It's another one of those corals. I think it's the same. I just want to see if we can get a nice shot of it. Yeah, it's another CF Anthomastus. So let's see, Maxie's on right now. Maybe Maxie can tell me if it's this Hellery polybus one. The sea cucumbers, scotoplanus. Awesome, thanks, that's good. No. Alright, we'll go ahead and keep transecting. Alright, there's something else she wants to zoom on. Oh, there's something in the sediment right there to zoom in on. Ooh. Ooh.
Looks kind of hairy.
It's like coated in. It is some sort of. Yeah, that's a cool worm. It's big. Oh, it's like sediment caught in its mucus. I'm not sure. Yeah. Yeah, some.
And if you see, like a struck that is
That's pretty cool. No, yeah, we're good with the picture. Awesome, thanks.
See?
Yeah, impressive. Cool. Onward we go. Okay, transecting away.
Anything else, Anna?
I think we're good. I don't know how evenly spaced these waypoints are. Let's see how evenly they space them in terms of distance. Oh, it doesn't say. Okay. It does look like, yeah, all of a sudden we have a lot of corals. A little habitat change again. Looks like some dropstones. A number of dropstones are here. We have crinoids, some sea stars. We have more bottle brush type. We have different types. Creminoid here. Single branches. Ooh, nice. Look at that star feeding. That's awesome. A big sponge coming off. Yeah, we've seen this a few times too, like attached to the end of a coral. Pretty cool.
Oh, who is that
Yeah,
guy?
Yeah, I don't. Do you know what type of worm it is? I'm not sure which type of worm it is.
The sponge or solus?
Did you see a solus? Oh, there is a solus. Oops. Can we? Sorry. Solus. Back up. That's on our. That's on our high priority watch list. Thank you. Yeah, probably we're gonna do a collection here.
Which one is that one here?
Sorry.
When that camera. Ooh, hold on. Let me see where I can.
Copy that.
I need to find it. Okay,
Here.
it's this one right here.
Here.
The cluster.
Yeah, she got there. She got
Where's that going?
Oh, look at him.
Let me see. It's. Chances are we'll section it. It's.
We'll favor the starter.
Yeah, I don't see anything else on the rock, so we'll probably section it.
Necesita tres.
Not seeing a request for anything else. I think when we sit down there, we'll zoom at some of these corals, see if I can tell any of them if they're different ones or not. This little pink blob right there, sticking out behind the oaf. Right there, yeah.
Por eso Patricia lo guardó en el cuerpo.
Your best bet is to bring that whole rock.
These pop like scratch right off the suction. They kind of peel off.
Star is a ophiopentaca.
Are you guys happy with the video so far?
Yes, we're good. So when you're
Barley cat.
So we're gonna get a solus, it's a type of holothurian or sea cucumber that kind of attaches itself to the rock and then puts out
You want to just put it on the ground?
You can just put it on the ground for a second. Clearly, Sebastian also needs 3 arms, just like the rest of us. Antarctic. Oh, I already forgot what he called it. Antarctic. Jason.
Oh, I got it wrong. It's Jacobfish.
Jacobfish.
I know it was some biblical.
Okay. I think you might have had it right, and I just have it wrong. Jacobfish. Yeah. It starts with a J. I just start coming up with J names.
No, I think it was.
Okay.
Okay, what do you want to
Jacobfish.
Oh, I've done it again. Jonas fish.
Jonas fish. Jonas fish. Okay.
J
Jonas.
names.
Jonas. Exactly. Let's
No
put
to
it
leave this.
in the. Let's go to 8. Yeah.
Pero sí hay que sacar la foto también. No, pero la foto para cuando se vea que está agarrando eso.
¿Qué esta?
No, sí.
Just gotta kind of scrape it a little bit. And it should come.
This is a cucumber.
Hopefully the rock isn't too.
Oh, I see it. In case anyone was confused, there's not the sea star, this is the cucumber next to it that we were just sampling just now. Well, yeah, because people like me, might be a bit confused.
Why don't you try the rock? It's a small rock.
Yeah, I didn't know it was that small when we started, but I think. Oh, yeah, I guess we're gonna have to just take the rock.
But Andres will be happy. He
pretty
wants
small,
some rocks for his geological research.
Yes. Is a rock by cats.
Cancel?
Yes.
Are you gonna label it a rock?
A rock.
Sorry. Usually they come off it. The rock isn't that small. I'm sorry. So.
Two cucumbers. You want 2 more of this?
Yeah. We can put it in the last quad for C.
Asking the rock first.
While we still have small rocks and put it in there. Yeah, we definitely have at least 2 species of rock there. And oaks. Here at UTC.
So the sea cucumber I think we've just collected is a very rare, special, interesting specimen for our sea cucumber specialist, and she's now very excited. But she wants two more. So now we need to watch out.
Otherwise they'll be very unhappy to be alone.
Otherwise the sea cucumber will feel a little bit lonely in its biological box.
Exactly. And the referees will say no. Statistically not.
4C.
So Erin mutes me.
And can we, yeah.
I said wait till Erin mutes me because which one is this? Bio box.
Oh, C. 4.
You know, I always
4C.
like getting bonus samples.
C. BB 4C.
Except for these are hope the people who are taking the bonus samples have enough materials.
The name in this.
Local time right now. Local time right now it's 10:15 p.m. Thank you. WB4C.
Thank you.
And then right. Alright. Can we grab a Niskin bottle before we go? And then actually I might have you guys grab a coral here too.
You want to grab the coral first?
Yeah, let's grab the coral. Sounds good. I want to zoom in these three corals here. I think
he's found them.
Oh, I told you.
Oh.
Okay, so that one. That one's kind of paired. Okay, this one is different for sure. Alright, can we zoom in on this next one to the right? Yeah, those two are definitely different. Okay, and can we... Ooh, I see nodes.
Sí, bueno, que la tenemos, o sea.
No, this is a bamboo coral.
See, this is a bamboo coral. I see nodes. Yeah, we definitely want that because we don't have it yet. And can you go ahead and go over to the right a little bit?
Bamboo coral.
This one. Yeah. Alright, we definitely want this bamboo coral here. That's gorgeous. That's great light on it. And then,
maybe? I don't know what is the taxonomy.
yeah, you can, yeah, go ahead and zoom up. Let me see if there's, what else is up there. Yeah, that one's another Thorella. Yeah. That one's also a Thorella. Okay. Yeah, let's collect the bamboo with the nodes in the middle and then the one to the left of it. Different quivers. Your choice on the quivers. Oh, I'm so excited. We have a little bamboo. It's so cute.
Yeah, it's small.
I like that its nose are kind of gold and sparkly. Someone was asking earlier if we find gold well. On this little bamboo, a little gold color. There's something in two. Yep.
Yeah.
Between here and Oregon, I think we're like a 5-hour difference. There's someone asking, we think 3 hours off from Central Time to Oregon Pacific Time, so it should be 2 hours. Yeah, like 5 hours before. So we're at night. Hold on. I read the wrong time. Sorry. What time is it right now? I can't tell. I have both times on my thing.
10:20.
I was right. It's 10:20 p.m. Okay. The other one is. Okay. Yep, the one on the left and then the one to the right of it.
When
Yep.
the coral in the box.
So this one we're collecting first. Thurilla or Primnoid? Pretty sure it's a Thurilla.
You want to cut it base?
Middle of the way is fine. Yeah, we can leave half of it behind.
Is it like big, this coral, for scale?
Let's see, the minutes are what, like
4 inches.
4 inches, so like a little over 10 centimeters, yeah. So, no, it's not a very big coral. It's probably like 25 centimeters or so. I just try to leave a little bit behind so it grows on.
Okay, you want to photo?
Yes, glamour shot, please.
That fish in the background?
Yeah, the UTC is, I'm confused, it's a few hours ahead of us.
4 hours.
Awesome. Oh, beautiful. Alright, thank you. These always have such small polyps, they're hard to get good glamour shots of, but it's nice. UTC. That's one. 662 meters.
CQ1?
Yeah, this one's going in CQ1. Yeah, it's very confusing, especially since my phone has 2 time zones right now. I'm a little bit confused on why my time. Thank you. And it gets a little deceptive here. That it's like always light out, right? So I went outside after dinner and after we had a meeting. I think it was like 8:30 or 9 o'clock at night. 8:30 because my shift started in here at 9. So 8 to 8:30 I was out there, and it's so bright. It's like 2 o'clock, 3 o'clock in the afternoon type of light. It's super bright. So it kind of throws you off, like then when you come in and then you have no windows like in here. So although at this point I've mostly been up since like 6 o'clock this morning, 7 o'clock, 6 o'clock, 6:30. Awesome. Yeah, one more and then a skin bottle. And then we'll transect some more. And they want to get to waypoint 4 before 4. But we're seeing some new corals, corals we haven't collected before. Another fauna, so. pretty fluffy. And again, about half of this one, so if you can leave the other half behind.
Got it.
You want half only, right?
Yeah, the upper half. Not the bottom half, not the right half. Yeah, maybe a smidge lower so you can get a glamour shot of the top of it. There we go. I think that's good. Yeah, these fluffy corals are like, I don't like to let the nips get in there very well. This is where you like start turning your head. Like. I'm sorry, we'll get the. Mm-hmm. Polychaete.
I think we cut it out.
Well, there's half a polychaete left to grow back to, maybe. Never know. Alright, nice. Thanks. That's good. Yeah, so this is some sort of bamboo. Something like Acanella. And put CF Acanella bamboo. You can see 126. Come on, bamboo. Well, half is better than nothing, I guess, huh? Okay. No, the sediment is definitely greenish. It's all the phytoplankton, all the productivity here. Lots. When you get up to the top, I don't know if they've been showing the stream up until we get to the surface or not, but like the water is just all green looking instead of the normal blue that you often see on these dives. Yeah, bamboos can be a little sticky, even these small ones. Not as sticky as, usually not as sticky as black corals though. Here we go. Just caught on. Use the force.
Shaky, shaky.
Take it. Almost. Almost. Got Maxie chiming in from home, saying it's Acanella, sending me a message. Yep. Yep. He sent me a whole bunch of literature for our next expedition today. to look at tomorrow when we're between stations.
You're supposed to put it inside.
Maxi dice que se parece a
Atenea, sí, lo vi.
que le mandaba saludos a Tyler y a Bel.
Yeah. Maxi says hello.
We miss you.
Okay. Tyler and Ben say hello. Ask where you're at. Awesome. Thank you. So those pieces are okay. I think we got enough in there now. Yeah. Thanks. Just like conveniently, it's in 2 pieces, so you know, Teresa can
Teresa can keep that part, just good for no.
Amazing. Thank you. So we can get back in transecting position once you recover, get squared away, and get this scoop. Oh yeah, Niskin. Thank you for reminding me. Yes. Thank you, Tyler. I asked. I just. So Niskin level 2. Side. That box. We have to go out there when we're transiting so you guys can show me how this fancy new deployment system works. You guys have made some serious upgrades.
You ready?
I'm ready. Skin 2. Went. Oh, that's cool. So it has to go in order. Oh, that's cool. I like it.
They're all the same size now, too.
Yeah. All the big ones? Yeah. Super. Every time I come back, there's an upgrade.
Something new, right? Somebody wanted it. So, is that somebody? Okay, shall we proceed?
Yes. Let's see, start moving.
Vamos.
Okay. Lasers are on, great. We're in zoomed out, transiting camera position.
Darby Bridge.
Let's see if we see another.
Taking off again. Is our current heading still acceptable?
It is very acceptable. Looks like the tides have turned, so it's in our favor. So yeah, looking okay at the moment.
Spectacular, thank you.
Cheers.
So yeah, I learned a fun fact today that I didn't know, that icebergs largely here move with the tides. So The tides are with us right now. And then we have sea ice. The sea ice moves a lot faster and moves with the wind, so sea ice is not our friend. It likes to chase us. And of course your sea ice is the ice that's forming from the seawater. And your icebergs are what's coming off the land. Nice. So pretty. No, we can keep going. We have to transit for a while, so we need to make it to waypoint 4. So
on the rock.
unless someone like puts in a strong demand, sponges aren't on our priority list.
For suction jars, use 2, 5, and 6.
I have 2, 5, and 6. Yes. And a failed attempt at 8. Yep. That's a crinoid purple, yeah. Crinoid, we're starting to see like these pops of yellow, some yellow crinoids and yellow sponges. Still seeing what looks largely like they're primnoid corals, kind of bottle brush or branched, but we see little bushier, like more fragile looking ones. Those might be the bamboos that we just collected, or the bamboo that we just collected. Ooh, is that a skate? I always forget. Ray? No, I don't want one. You guys have,
Yes.
they're gonna like fire me if I don't keep us moving. There it is. But we need something like this for the media footage.
That is nice. Our fishman is here. Happy.
Yeah, I'm really bad at telling them apart. People are like, it's so obvious, and then I forget every time after they tell me. Nice. The lasers on it, and you can pop off the lasers and do a glamour shots for the highlights. There we go. Look at that, like, all that, like, flocculent, like, material that rained down all around it. Just like,
Oh wow.
It's getting annoyed. I don't see any spines on it, just looks prickly all over.
Nobody going to?
all right, thank you. Lasers back on, transiting position, onwards we go. There we go. I'm just gonna check the paperwork here and see. You can keep going. We need to transit for a while. Yeah. And, we need to get to waypoint 4 probably in the next 45 minutes or so. So that we have time to do the push coring at the top and see what's up at the scour. Unless I hear otherwise, I'm understanding that they want to wait to do the push cores until we're at waypoint 4.
Are we
or
doing
further.
the push course on our shift? I thought it was next shift.
No, I think, well, next shift, but I thought they wanted it after waypoint 4, is my understanding, once we get up to the ice scours. But until I hear otherwise. Yeah, but I think they're gonna hit them. So have like an hour. They'll do it for the last hour of the dive, I think.
Yeah, I think Pedro wants to choose where to get paused from, maybe.
I think so. That was my impression. So unless I hear otherwise.
It'll take us there in about 40, 45 minutes to get there if we go 0.3. So,
Okay.
if you want to get there 45, we gotta.
Yeah, just translate. Yeah, just transfer it out. Yeah, like they didn't give me the distances between the waypoints, so it's
Hard to judge, right?
It's hard to judge, so okay.
Okay, we're not stopping now.
Yeah, we're transiting for the better part of the next. Wow.
Everybody pee before we leave, because we're not stopping.
Exactly.
Pack snacks.
Yeah.
But we still want to stay within transecting speed, so we don't want to go over the ROV speed of like that 0.3, 0.4. Okay. I mean, if we're a little, I'm kind of setting a slightly earlier goal.
Yeah.
It'll give us a half an hour on our shift to sample at waypoint 4, basically.
Okay, yeah. So this is pretty good speed. That should start a little here. Bridge.
Okay, perfect.
Bridge, alright.
Later.
Why, hello there. We are going to try to make a beeline to waypoint 4 going about 8.8 for you. If that works.
Alright, yeah, let's do it.
Let's do it. Fantastic.
All right, so we've set our speed. Yeah, we have quite a ways to get to waypoint 4.
600 meters.
Yeah, we've kind of seen these small changes in habitats, and then they kind of change, or communities, and then they change back. So we've seen like coral dominated. We're back to seeing here with these irregular urchins and some sea pens and holothurians. We've passed areas where there have been a number of dropstones.
You need a coffee.
You can squeeze it in your mouth. Straight.
It's just getting late in here.
That coffee needed, I told you.
Maybe we should bump up the speed a little bit now. Yeah, it should be
Yeah.
4 now.
Oh my gosh, this is strong smelling. Let's see.
You try?
That's
But it's not dark chocolate. But because
It's okay. It has coffee in it.
Okay. Do you wanna try one? Can I borrow just give you some flour?
Oh, yeah, absolutely.
Lista? Okay. Some coffee.
Oh my gosh.
Coffee.
Muchas gracias.
Yeah. Coffee filled chocolate.
Coffee.
Amazing.
Coffee. No?
Espresso.
Wait for tomorrow. Thank you.
Get there, like the last few we've been collecting are kind of purplish, but that one looked more white. Very possibly just a color variation in the area. But collect one later. Carnivorous tunicates, yeah? Will it be Maritza?
What? Tunicate.
¿Piensas que eso es un tunicate? No, we're good. No, we're good. Or no, it's
No.
yeah. Let's just stop and look at it really quick. I can't tell what it is actually.
Yeah.
What I thought it was at first, I don't think it is. Yeah, this like balloon looking. Oh, okay. I think it's a type of worm. Maybe. I don't know.
I don't know. I think it's Jenny.
What do you think it is?
Careful with the baby. There's a baby also good.
Yeah, but that is
Very small.
I don't know.
Yeah.
Decision. Will collect or
No, we're gonna go. We're just gonna. This is good. We're good. Yeah, we got enough of it. I think someone else can identify what it is. I'm sorry, I don't know for sure what it is. I feel like it's like it's feeding apparatus coming out of something, but I don't know what. So we need Maxie.
Where is Maxine?
That's alright. It's something I haven't seen yet, so maybe someone will. My first thought was a tunicate, but now I'm not so sure. So
Wait, I'm searching. Let's see.
we're gonna try to search and see what we can find. Oh, that thing. think it's not.
Did we pass it?
It was an Ascidian.
It's an Ascidian.
Okay, that was my initial thought, was some sort of Ascidian.
But he had something here.
Yeah. No, we can keep going. We can keep going. We're just trying to identify what it was that we were just seeing. I think it's this or something similar.
Yeah.
Dystaplia. Dystaplia. Looks like it's a dystaplia. That book is lovely. So yeah, it is an Ascidian. So my first instinct was, how do you spell that again? Dystaplia. So that was something I don't think we'd seen before. Yeah, a little rock with some corals. Oh, sorry.
Was there? The solos, no, but they are on the rock.
Yeah, but it was not there.
Yep, so I was right at first. Thought it was a tunicate. Looks like it is a type of tunicate. Okay. Sometimes I just need to go with some anemones. Sometimes I just need to go with my first instinct. And then I second guess when it's not my field. Well, there's a sea pen there.
Next station.
Yeah, we've been seeing some on belugas. I think we just passed one a moment ago. So I think it's probably in the genus Dystaplia for that tunicate we just passed. Distaplia. Are we changing? Yeah, we zoomed out so we can. Yeah, make sure. Did our slope just change or something? Seems like we're more zoomed in than normal. Okay.
It's sloping up a little bit.
Okay, that's fine. Just want to make sure we're in this correct zoom position for the transits. Thanks for checking. Okay. Yeah, it's supposed to get closer to 500 meters, I think, for this. Waypoint 4 is, well, actually, waypoint 4 is only going to be still 660 meters. I don't know. I think it's just their text doesn't match the current dive plan. I think it's from an old dive plan. So it happens you have to change things last minute. Alright, so yeah, it looks like we're going to get to only 660 meters. What now? I think so. Like, it's definitely why my phone wants to autocorrect spelling of scientific names to other words that don't make any sense. Like they're also not like words. I'm trying to message someone to check. Fishy. Sea cucumbers. Every time I see rocks, they tend to have these anemones on it. Crinoids, some corals. sometimes some sponges. Occasionally we're seeing solus. Not too many though. Little benthic jelly floating by there. At least I think it's a benthic one, just a little further off the seafloor. Oh, there's a nice, is that a what is that called? Serianthus, yeah. No, we can keep going, it's good. We've seen these a few times on the dives, just this one's a good size. And there's another one of those tunicates down there. A couple of them. Ooh, pretty shrimp. That was a great shrimp. Yeah, Serianthid is what we just saw. And it was quite a good sized one. I haven't seen one that size on this expedition yet, although I haven't been watching all the time. A number of those irregular sea urchins here. Sea stars, tube worms. Huh. Well, our book was suggesting it might be dystoplia, but maybe hydrolina, and that serianthid we just saw was a branchioserianthus. Or the hydrolina was the branchioserianthus. I'm trying to interpret the message here. But maybe what we thought was the Staplia maybe is a different genus of tunicate, because that one's colonial. But it really looked like the picture in the book we have here, so we'll see. We'll do some looking later online, see if we can narrow it down. Nice sponge. Yeah, so the branchioserianthus. Always trust Maxie to correct me. I get it narrowed down. Yeah, we saw one of these earlier. I think it's a sea cucumber, but I'm not sure. Or no, it's a Tina. Is that a Tinophore? It's a benthic. It's a benthic Tinophore. Yeah, it's a Tino. Now that I can see the angle better. Yep. Pretty Tino. Yeah, that's true. A lot of books have errors in it, but there isn't a lot of information on this area, so it could be a little complicated. That's similar to something we collected, but I don't know. I just don't know. Yeah, I don't know. That looks like a but that's a hydrozoan though, not the right type of polyps. Yeah. There's another branchioceranthid. Brachio. Is there an N in there? There's an N in there. Branchioceranthid. Lots of these holothurians that we've collected already. Sprinkling of these primnoids here and there. I think we've collected most of the species that we've been seeing. It's hard to tell because you have to zoom in or have them in your hand to tell for sure. Let's see, which one of these levers? Oh, yeah. That's better. Sorry, the microphone noise is going to be weird for a second. I just have to adjust it. I sat myself up higher. Alright. I've been seeing some of these urchins as well. Something similar to Stereocedaries or Cedaries. I always forget which is which in my refresher course. The big anemone with the orange tentacles might be an Ormathidae. We're doing shift change for the ROV pilots.
We're not stopping all the way before?
Unless we're stopping occasionally to do a little zoom. We're trying to stay around 0.3 meters per second, about this height. And yeah, once we get to 4, we can kind of do more collecting again. It's just waypoint 4 was a lot further away than I thought. They're not evenly spaced, so but our goal is to make it the waypoint 4 and beyond to do the rest of the sampling for today. Okay. Okay, we'll do. We'll try to keep our eyes peeled and not try not to tell you at the last minute too often.
We've known to come back. Thank you.
Yeah.
I appreciate it.
Hey.
Hi. A little note.
Yep.
Where did we sort of get to on the
Are you on the lead next?
Please don't.
Yeah. So what did you want to know?
Just sort of where we got up to. We still got the push coring ahead.
Yeah, we still have the push coring, which I guess Pedro wants to kind of select, but I know they kind of wanted to get to waypoint 4.
It's like a zoo.
So
yeah, we're transecting straight onto here.
Yeah, so we're trying to get there because I think like the waypoints are a lot closer once we get up there.
Explodes in your mouth.
I think 5's the last, isn't it?
No, it's to 7. It's all the way to 7.
But that's all points.
But it's all like the iceberg scour marks, so they wanna I think sample up there. So right now we're seeing a lot of what we've been seeing, so it's a good time to transect anyway. So we're just gonna do that, and then maybe once we get to the scour marks, try to do more sampling. But yeah, I think they didn't update everything, but I was pretty sure from the meeting they told us 2 o'clock.
We were heading 2 extra hours.
Yeah, so I'm just trying to get us to waypoint 4 by like 11:30, so that we have like a good half hour to do some sampling before they come on. Well, you guys come on, and I think they'll want to push core unless they tell me sooner.
We're not changing now?
No, we
change
change
it
it.
midnight.
We change it midnight, so we're good.
Okay.
Yeah. So
well, thanks for the update anyway.
Yeah, let's get you. Yeah. So that's what we're doing right now. But yeah, we're on 3-hour shifts. They're on 2-hour shifts. So.
Right. Oh, we staggered everything an hour, didn't we?
We
as well?
staggered everything.
We extended and we moved everything an hour.
Yes, we did. So it's a little confusing.
Thanks.
All right. Yeah. No worries. Thanks. Yeah, we had a little shift schedule change today. Like our group should have been on a couple hours earlier, but because we came out of the water and stayed out of the water longer, we just all shifted. So we're diving later than we thought, and we don't have coverage for everyone at night, so we just shifted everyone later. So everyone's just a few hours behind. So okay. Oh yep. Sorry.
Bottom left.
Sorry.
Next Mollusk, if you can find it.
I'm really sorry.
Who's the Mollusk for? For who?
Shout stop.
Leslie? Okay.
Yeah, don't do so much other talking because I didn't know that. Yeah. Or quieter, and then they'll jump out at me. Okay, sorry. We had a Apparently they spotted something, but but there was so much going on I didn't hear until and we're going at like a set speed right now, so it's kind of hard to slow down. So okay. So what was it that they saw? Was it a mollusk? Okay. Okay. What color was it?
White.
Okay. I'll keep my eyes peeled for the typical white gastropod. Alright, back to focusing on the screen so I can catch anything. Okay. Nope, there's another one of those Anthomastus over there. Arthurians. Ooh, that looks like a different urchin than we've seen before. I think it's an urchin at least. If we see more of them, we can check one out.
So when we get to weapon 4, then we stop taking some push course.
Eventually, yeah. I think for the first half hour, we'll kind of just slow down and look around. And ooh, there's another one of those worms we saw earlier. Another Anthomastus. And then, yeah, when we get there, we're gonna, I think, kind of slow down and look around and zoom in on things and maybe do some collections. But I think around midnight. Where? Stop. Stop. They're seen. All right. Back to the left. Okay, back to the left.
Wow.
Left. Now.
Yep.
Yeah, I see the corner.
To the left. This?
Yeah.
Okay, that looks okay. We can zoom in it. It looks.
I think this guy.
I think so too. But hey, let's take a.
He's alive.
Yeah.
Lobster.
Shrimp. Do you still want to zoom on it? We go ahead and zoom on it anyway. See if there's. Yeah. Okay.
It's dead. It's dead. Yeah, it's just a shell.
I like that things, please.
The dead? I like it here. No, come on. It's dead. Yeah. We can go for the next one.
Okay, we can go ahead and go. No, we can keep going. We're good.
Yeah.
Alright.
Yeah, we want live ones, so.
I like some dead ones, not gastropod, brachiopods.
I like brachiopods.
You like brachiopods?
No. Yeah, no, mollusk.
Okay. Keep looking, see if we see one that's living, but we'll transect mode with the zoomed out lasers on we go. Right now again. Are we zoomed out? Yeah, out like this. Yeah, we want to be out like that. Thanks. Sometimes I can't tell if it's just we're steeper or. Okay, thanks. That's the designated transect mode is zooming the camera out. So for this expedition at least. So how long have you been an ROV pilot? 22. Did you work in?
It's only my second year.
Yeah, second rotation for Fall Corps 2.
Yeah.
Can we go about 0.3 on the ROV speed? Yeah. So have you done a lot of science?
Not really.
Not really?
Oil and gas.
Most of oil and gas before?
Construction. Survey. This is the first time I don't want to have many years.
Okay.
Most of the time it's just...
Nice. Yeah, okay. So So you're getting used to all the weird requests for fauna. Scrape this organism off that rock.
We don't have that privilege in oil and gas. How we want to look at some stuff.
Yeah, it's pretty special. You get to learn, like, okay, this one's squishy, this one's sticky. Yeah. Maritza, do you want to tell them something about sea pens? Lulas.
Hola a todos, aquí Maritza Castro, bióloga marina, y estoy intentando especializarme en plumas de mar. En los cruceros anteriores en los montes submarinos encontramos 10 nuevas especies para Chile. Aún necesita ser verificado ese número, pero lo más seguro es que sean 10 especies. Y en el caso de la umbelula que pudimos ver anteriormente, que es como un paraguas casi, al parecer tenemos 2 especies diferentes, así que dentro de quizás 1 o 2 años haya ya los reportes normalmente serán oficialmente hechos, ya que esa va a ser parte de mi tesis de quizás magíster o doctorado, así que ahí estén atentos a las redes de SMOI, que es el Centro de Ecología y Sustentabilidad de las Islas Oceánicas, con el cual trabajaremos este paper.
Yeah, so Maritza is our expert in sea pens on board, and she recently graduated with her marine biology degree, or an undergraduate degree, and she's hoping to do some master's or PhD with sea pens. She wrote her thesis, did an amazing job working on a publication, so yeah. She's been on, I honestly don't know if there's a scientist who has been on more Falkor 2 expeditions, or Schmidt ocean expeditions in general, especially in 13-month period. She's been on one more expedition or one more leg than me at this point, so in the last year. So she spent something like 5 months on the ship in the last year, going on 6 now with this one, I think. And yeah, for anyone just joining us, we are on our 3rd dive of this expedition, dive 777, and we are doing, this expedition is the Climate Connections at the Icy Interface Cruise. We're off the coast of Antarctica and the Bellingshausen Sea, and I think the Ronne Entrance, we're at our first station of this expedition. We have a number of people in the room with us right now. Let's see, we've got MT Larissa is over there kind of doing the streaming work, and kind of Andy and Joelle piloting right now. Joelle's flying. Andy will do the sampling when we get to that. We have Anna in the room, Maritza, Sveta, Teresa, Jessica, and Tom. I don't know, Jessica, you haven't spoken yet, have you?
No.
You want to? No? You don't want to?
Yes, you can.
Okay. Well, Jessica's here helping out Teresa, who maybe you heard earlier. Is working in Portuguese. No. She's not ready yet. It's her first cruise, her first expedition, so hopefully she'll want to speak up soon. And we got Maritza, we have Sveta and Anna, Tom, our resident fish expert. So we got a number of people in the room right now. The science team will do some shift change in about, let's see what time is, about 45 minutes. And we're working right now up this slope to around 660 meters or so, where we're hoping to see some ice scours based on the mapping that was done yesterday. Expedition the scientists on board, there's 24 of us, and we have a range of expertises from oceanographers to geologists, geochemists, biologists who focus on the animals that live in the sediment, to biologists that focus on the larger organisms. We're a little over 100 meters away from our next waypoint, so we're trying to transit to that waypoint, and after that is where we expect to see the start of a large ice scour.
What's this mushroom over here?
Which one? That's an see if Anthomastus there. It's like a mushroom. That's an anemone. Yeah. So the orange one is an anemone. The purplish one is a coral. A sponge off on the right. Lots of holothurians or sea cucumbers, also known as sea pigs. We got some tube worms out feeding. Where? Oh, they're down center. All right. Well, sorry we missed the Octopus Friday. Tunicate recently that we weren't seeing earlier on in the dive, at least that I'm aware of. Not during the first hour and a half or so of my shift. Okay, gastropod, there we go. Right there. Thanks for spotting it in time. This one might be alive. I think it is.
How can you tell if it's alive?
If you see it's like foot. Yeah, let's go ahead and try to collect it. How? Yeah.
Tell the audience.
Especially the jump.
Bridge, RV, can we pause and move, please?
Pausing and move.
Thank you.
I think I see it. Yeah, I think I see.
But that I think is of you. But let's see. Yes, it's of you. It's enough you.
Yeah. I don't know, the shell looks kind of broken, but I mean, maybe. Just give a zoom on it and see. We might have to poke it.
Yeah, let's see. I mean, the position is very good. Yes. It's like, yeah.
Oh no, that's just like an O farm laying on it.
Christian wants us to take
Yeah,
it.
I think we need to. Do you think this will fit in the suction? It's small enough, right? It's like 3 centimeters across.
It's like a line.
It's 10 centimeters. I'm only 8, but it's nice.
That's narrow enough, yeah. So do you think we could suction it, you think?
I will try.
Yeah. I don't want it to get
We
stuck.
can
Yeah.
scoop it also, whatever.
Okay.
Okay.
Let's try to suction it into like 8. I think it was already on 8. Oh, okay. Yeah, you can try the scoop. I mean,
Try the scoop?
if you already have the scoop in hand and it's easier, you can try it.
I'm
Andy,
gonna
what?
have to regrab it anyway, I just got it.
Alright, let's just try suctioning it. I'd rather suction because it's easy to break mollusks
Right.
sometimes.
Suction it.
Yeah. Sorry for my indecisiveness.
Suction on.
So in my opinion, we have this
Number
really
8.
cool Hoover, underwater Hoover.
No. Just wait.
Yeah, it's definitely alive.
That is going to number 8, alright?
Yes, number 8.
Yo creo que es mejor con la... Puede ser que se pueda atrapar en... Ya. Ah, no.
There it goes.
Gone. Thank you.
Shaky, shaky.
Yeah.
Yeah, I was waiting for it to pop, fall into there. There it is.
Yeah,
Yep, it's in there.
Ah, no, pero es...
And ocho.
221. What is it?
And ocho. Yeah, the foot was definitely there. Yeah, it's definitely. Yeah, it's just sitting at the base of the coral. It might. Some just live there, some eat the coral. So. Awesome. Thank you.
In the lake, at 40.
What is it?
Back in transect mode. So lasers on. Zoomed out. 0.3.
Necesito comprobación de que explicación. No me crees.
Aphoria. We said that the genus is Aphoria. For that mollusk. Ooh. Nice. Cool. I think my mollusk spotting eyes need a little practice. I got so used to it for Javier, but oh, there's C-pen.
Bridge.
Hello. Yeah, so we got that updated waypoint. and that
Waypoint
direction
4.
we're on the moon,
Updated waypoint, thank you.
It's amazing, like you do this for just even a few hours and your eyes start to get trained to spot things. But after a couple days of it, you get really good at it. But after a few months away, you lose it. You have to regain it. anemones. Jelly. Thank you.
How many samples you collect?
Look at all the urchins. So we're getting an area with slightly more dense population of these irregular sea urchins. Still seeing a few corals from nodes mixed in. It was in 8, yeah.
No, that's up in 7 as well.
No, we don't. You can index a 7, you can leave it in 8. I don't think the mollusk is gonna swim away, so. Yeah, you can leave it on 8, I think it's fine for now. Yeah, sea pen, there's a little anemone on the sea pen. Almost to waypoint 4. I want to see this iceberg scour mark.
To be honest, iceberg scour marks are normally like really large scale features, so I'm not sure if we'd be able to see them with a camera like this. Like they're like really big things, so
yeah, but we won't see like any. Do you think it's mostly going to be covered up in sediment, so we're not really going to see the exposed rock?
Still
Okay.
going to just the sediment area, but there might be some like ripples, you know, on the sediment or something maybe.
Okay.
I'm not sure.
Let me see.
So they're very large, and we cannot.
Yeah, nice sponge over there. Yeah. Do you see earlier? We righted a sponge. It was on its side.
We're going to see like small.
We wanted the polychaete and it went to the underside, so we're like, just pick it up, straighten it up. That was nice.
You have to ask Sasha.
Yeah. Well, see, I'm learning things all the time. I didn't know, like, I mean, I kind of figured they'd be big, but I thought we'd see, I don't know. We'll see. Well, either way, I'm excited to get to waypoint 4 because I feel like I can collect again.
Yeah. We are waiting for that moment. Porque sin pink.
Fish sleeping over there.
What here is actually the scholars as was the weather sea. Weather sea was -1,
jellyfish.
7.
Yeah.
Was that?
Weather sea.
Is that where you went on the last expedition?
Yeah.
Yeah. Okay. And what's the temperature here?
1.6.
1. Okay.
Now it's positive 1.
Positive 1.
Minus.
And it was -1. Either way, it's really cold. I was thinking it would be a little bit colder. Like I thought it would be minus or like pretty much right at 0.
Actually, I thought this area would be way colder than the one. It seems like it's actually cold. Yeah, it looks like not as cold as weather sea.
Thousand, that might be much colder, especially when you go deeper.
Okay. Octopus. Can we zoom in on the octopus?
Oh, you want me to stop?
Yeah. Sorry. We gotta get our media highlights for Alex. And plus, the whole world loves to see a nice little octopus. missed one earlier, so.
Octopus?
Mm-hmm.
Well done, you missed the video.
I listened to Erin.
That's why she has the headphone.
Yes, that's a pretty one. It's cute.
Well, that's cute.
It's like 10 centimeters, like 8 centimeters.
Yeah.
There's no one in our science party interested in collecting octopus?
No. Mm-mm.
I don't collect them.
I like how it's like looks a little iridescent there, like when it's like the spiral, how its legs are spiraled.
Sleeping?
Yep, sleeping. Onward we go. Hopefully that's enough for highlight.
Okay. Jill?
Yep. Yeah, this is I think the first one we stopped at. We saw one earlier, we just couldn't. I saw it. We saw it as it was going out of screen. So I was too full on backtrack to stop and look at it. Alright, change it to 10 meters, 10, 20, 30, about 40 meters left to waypoint 4. Yeah, I feel like it's been a pretty interesting dive for like diversity of fish, and I haven't seen too many fish lately, but early on we were seeing quite a few fish. I'm still seeing some, but not so many swimming.
And then once we get to 4, do we keep sailing to 5 or we just stop?
I think we can just feel a little more free to like stop and zoom at things. They want to make, I mean, they have to 7, but the instructions aren't really clear if like, I think they just really want us to get to like the 4 to 7 area, but 4 and 5 are quite close comparatively. Like 3 and 4 were really far apart. Like 700 meters apart. Or 700, 800, which is like a long ways to go.
Here
So.
they say 3.4 to 5 minimum interaction. That means we can.
Has the vessel stopped or no? We just lost
No. They're not supposed to stop. They're moving according to the ice. So I give them the general idea that
Oh,
go
right,
this way,
right.
and they're going to do what they have to do. So
Okay.
Earlier we stopped long enough collecting a sample that the ice that was in the way had passed us. It's like, yes. We're like, we can't make it to waypoint 3. We're gonna have to go this way or this way. Yeah, another little octopus. You can. Dumbo, okay. Yep, it's a. We gotta zoom in on this one too. Do you see it? I'm telling you, half the time when you start seeing octopus, it's you just there's almost never just one. It's like you hit a zone. At least that's how it is on a lot of the seamounts we were at. Yeah, we might want to sit down and look at this one. 'Cause
Okay.
it's a Dumbo. Yeah. We're close enough to waypoint 4. We need a nice glamour shot of an octopus for Alex. It's Dumbo sleeping. So cute.
episode, does that?
And look, we have someone in chat who knows their cephalopods, so they can help us out here. Hopefully, if we get them enough information with the Zoom. I don't know, because it's sleeping, so I don't know how much they can get, but... Because it's so productive here, all this rain and material. Like everything we're seeing is through a green snow. There we go, it's clearing up a little. Sleeping. Just imagine like a Dumbo snoring, like, and you see it breathing, like, or what looks like breathing. Right, so yeah, why not? Well, I'm always so bad about this. So cute. Okay. All right, onward we go.
Okay.
When everyone's ready, got their pictures?
Yes.
Okay.
We have a serious.
All right, lasers back on for transit mode.
We need work.
It's also like surrounded by there's some like a lot of polytherians there. Fed through that zone. Earlier we saw a squid like dart itself into the sediment. It was like hand. It's been an interesting. Ooh, we're seeing a change in habitat. So quick. And this is right when we're coming up on waypoint 4. Ooh. I see like a little hill. I see topography for like the first time. This is nice. And there went the diversity. There went a little coral garden. It was there for a moment and then gone.
That's maybe because there's loads of rocks and stones and then
Yeah.
force things for them to attach to, and now we're back to fine sediments.
Yeah.
So you want us to stop, right?
No, we don't want to stop. We'll turn with the transect line. It's just now that we're at 4, I won't feel so bad about stopping and looking at things.
Oh.
So, because we had like this goal to get to the waypoint 4 to 7 area, because that's where there's some ice scours, and the people who want to do push cores want to do them up at this higher point. So, yeah. So now that I've made it to the goal, then I can. If we see another like different diversity like we were just seeing there, maybe we'll stop and look. So we'll just kind of follow their track. Ice scour, SCOWER. So like, you know, like, hold on, not W, it's an OU, isn't it? SOU, thank you. I can't spell, it's middle of the night. SCOUR, scour, like, you know, I don't know, scouring dishes or something, scouring pans, scouring pad. So it's like when the ice comes and scours or scratches across the seafloor. SCOUR. Yeah. No, we did dive 776 earlier today. Didn't we? I think so. It's written on my sheet. We're on 77. We're in dive 777, right? That's what my sheet says.
Yes.
Okay. Yeah. We did. Let me see. We did 3 dives at this location, and a short aborted dive 775. One earlier today. That was 776 on the left. Yeah, we can go ahead and go a little bit to the left and see what's on the top over here. Everyone in the room. Yeah, I think it's a yellow sponge, and then yeah, that's an anemone, and a polychilid, I think maybe is what that's. Is that a polychilid right there? I don't know what that is. Can we look at that really quick?
Can we just
Actually, there's a solus. Yeah, we can collect the other solus for Teresa. But not this. Oh.
Maybe.
Oh.
Bivalve.
It's a bivalve, yeah. Okay. Yeah. Not at all what I thought. Yeah, we're gonna do a couple collections here.
Sample I showed in the biology exam yesterday.
Nice. Yeah, we're gonna collect this here, and then we'll go up to that rock and we'll collect the pink thing on the side of the rock. And we can do both of those, I think, with suction.
Where's the one for Teresa?
It's up here on the rock. So we'll do this one first, because I think we're in good position to get that. We can get it with suction, and we can put it in full-size sections, so 7.
7.
Yeah.
That's 7.
Ready, Jarvie. Pause and move.
All right, I'll stop.
Maybe CF spondylus for the Bible? I don't know.
Spondylus is not
But
with a rock.
yeah, I don't know. I'll wait for an expert to like. Oh, it looks like Christian is chiming in. Okay. I think Christian's responding. He'll give us an update here. Oh, it's a shell of a scallop on a sea. It's on top of a sea urchin. It's a decorator.
Oh, this red one that we just found?
No, I don't think so. Yeah, no, we can skip this one then. I think it's a shell on top of a sea urchin, so it's not actually one. No, we'll skip and go to the solus. Sorry. Yeah. That's awesome spotting. I did not see that at all. Thought it looked weird. No, we're going to cancel this collection and go instead to the rock. But we're going to do suction sampling with the rock. But you have to pick up the scoop, so I don't know.
And are we doing the
Whatever is
sea
easiest.
cucumber?
What rock? Whatever rock.
No, on this rock here, there's something on the side of it. I'll show you on the screen. Right. Oh, it's hiding. We're in a bad angle right now, but there's somewhere on this side is a sea cucumber that we want to get off of there. So I think we need to rotate over. It was there. It doesn't move very far. It sticks to the rock, so it's still there. For Teresa.
Teresa says she doesn't see it.
I know, but I saw it. It's there.
Alright, so I can move. Maybe.
Yeah, move a little bit, rotate to the right, yeah. Or looking down counterclockwise.
Okay.
Ah. Okay, we will do the next one then.
Oh, they.
Ah, ¿la han agarrado el scoop? Have you checked the scoop if they took it? Yeah, there is no scoop.
I'll let
there.
you know when I see it. It's gonna be.
Look at there.
I think it's that pinkish spot tucked underneath there.
Okay. It's there.
Yeah, right there. That's it right there.
Yeah. So we have to scare the animal, disclose it, and then here it is.
Yeah, so this right there, and you should. I think this rock is big enough, especially with the anemone on it, that if it's stable enough, you should just be able to like suction it off by just hitting the lip of the red part of the suction thing and peel it off and suction it up, so you can put it into 7.
Far away.
Looks like maybe there's some stylosterid hydroids around.
Somebody else.
Steady?
Steady. Go for it.
Let Sonny get in there.
Oh, that's right, it shift change, huh?
Ready?
Alright, so it's shift change for our RV pilot. So we have Sunny coming in for Joel. Thank you.
Thanks for the company.
Great.
Hmm? This? No. It was here when I came.
Okay, leave a note because they have to leave the bottom at 1:15. They have to leave the bottom.
At what time?
They want at 1:15 because it has to be on deck at 2 o'clock.
Yeah, they know. Yeah, they should. They'll calculate it. Yeah.
No, they just can.
Okay.
They.
Hold on. Let me watch what's going on and then.
Yeah, no.
Okay.
Sit there.
What did you want to get, Aaron? This right down here?
Yeah, that right down there. Yep.
No.
Suction is on. Yeah, right now, that's good.
Look at that.
Let me see. Okay, so
Give me a little bit of suction.
And
Oh, he's really stuck.
yeah.
yeah. You know, these rocks are keep moving. Ooh. Not big enough. Oh, there it goes. There it goes.
Well done.
Nice, Andy. Alright, watch it go into 7. Solus, I think it just dropped in.
No, it's still in
Oh, is it still in there? Oh, something dropped in there. If it won't go through the suction, you can drop it into like a coral quiver if you're able to pop it off with the other arm.
Yeah.
Let's see.
So,
I got it
or if it's easier, actually, you can just dump it on top of. You can put it in BioBox 4.
4?
Yeah.
Which order?
Yeah, you can put it in BioBox 4B. You can drop it in there.
Oh,
because, yeah, probably.
Yeah, 4B.
4B.
Okay,
Boy.
No, it's still on. Top.
There it goes. There. Yeah. Great. Thank you.
In. Thank you. Yeah.
4B. 4B.
Yeah, Quattro Bay. No, we're good. Sorry, my thing. No wonder I can't hear you very well. My one side of the ear headphones fell down over my ear again.
So, it's missing just one.
Alright. Amazing. Thank you, Andy.
Is there more sampling to be done here?
No, we're good. We'll continue.
If we landed, do you want to try for the fish? Can we reach that with the set?
You think it's gonna hold still? Do you wanna?
It might go off like a rocket.
Yeah, I mean, we can try suctioning it if you want. Keep it on 7. Sorry, Andy. Can you try suctioning that fish? Are we in a position where you can do it, or do you have to shift over?
It's pretty far out. No,
Wait.
don't worry about it. It's just what we're already landed.
Okay.
I think it's going to go off like a rocket once soon as we get close.
Okay. All right. Never mind. Let's go ahead and move on then, and if we land by another one. I think we'd be better off with a net, but yeah, let's just keep our eyes peeled for like.
Just a particularly nice species usually caught online on long lines, so they end up with a jaw damage.
Okay.
It's just a particularly handsome little.
Okay, it looks restless right now. It started to
it
move.
already looks wary.
Okay. Yeah, let's go ahead and just continue. Yeah, it's already moving on its way. We'll see if we can sneak up on one later. Yeah, so we'll just go ahead and head back towards the transect line, and lasers are on, transecting. Yep. Looks like we've got some nice dropstones here. Yeah, so we have some people on board working with the holothurians, or the sea cucumbers, and Solus is one of their target groups that they want us, the rest of the group. So I'll let the documentarians pass on information, and then I'll switch over with Tom here as we head on this transect. All right, Tom, you ready? All right, see y'all in a couple days.
For something, you like some of biology or geology? Okay.
Just gonna start around.
Close another one.
Extend it.
Better. Pushkar and Saki, box, bio box. Okay, let's go. Aquí, geology sample.
Need a mic as well. We should have a chat. Hello everyone, that's us just transitioning between our science teams. This is exciting, but it's quite exhausting actually to maintain this level of focus, so we try and roll people out. Also gets the different set of expertise as well. So I'm Tom Lindley. I am a fishy person over from the Museum of New Zealand, and with me is... You ready to say hi, Pedro? Yeah, introduce yourself. Yeah.
Yeah.
Do you want to say hello?
Okay, hello. My name is Pedro Martinez, and we are going to take the last shift this night. We have something to do. We have a lot of work to do. Let's see how it works. I'm from Spain, but I'm working in Germany already for some time, already 30 years. I'm working at the Senckenberg Institute. This is a research institute, and we have also a very nice museum in Frankfurt, in Germany. And we are dealing with biodiversity. Not only marine is one of our specialties, but we have also specialists dealing with all kinds of organisms, with birds, with mammals, also with fossils, and botanists. So it's a large institute. So now we are here in the Antarctica, Bellinghausen Sea, and we are trying to collect some samples in order to study the biodiversity here.
So we've just come up on a couple of dropstones. That looks interesting. Are these large brittle stars? Their arms up into the current to filter feed, maybe? And on this first one, there seems to be a collection of a few different types of sponge. Sea urchin just in front. Yeah, let's zoom into the first one. Cool. Is that a bryozoan at the front?
Yes,
the
this
white
seems
lace.
exactly. This seems like a bryozoan on a sponge, and then we see also a crinoid, beautiful one. And you have seen also the holothurian, right? The sea urchin.
There was a little sea urchin, yeah, just in front there. Does anyone have any interest in the brittle stars? That I'm not sure we've seen that large filter feeder at the back.
For the moment, I think that nobody have claimed interest on the brittle stars.
It looks pretty big. Those laser scales are 10 centimeters apart, so that is 20 centimeters across, I'd guess. That might be quite a wrestle to get that down. Okay, if we continue along the transect.
So we need to leave the bottom at 1:15. This means we have a little bit more than 1 hour to go. We should think in taking also the push course.
Yeah, we should probably prioritize the push course.
I think that's about 18 push course. I think that's what we need to take.
Okay.
And whenever we find a good spot to land, maybe we should then take the push course.
Did we have any priorities beyond being into the scour area? Are we looking for any particular sediment type?
Well, no, it could also be outside the scour area where it is more flat. So more in the greenish area on the map. Yeah.
So waypoint 4, which we've just passed, we're on our way to waypoint 5. That marked the beginning of the iceberg scour. So that's where icebergs have made contact with the seabed in the past and have sort of trawled through it, but that's at a much larger scale than we tend to observe things, so we might not even be aware of them. We might see little areas of dropstones where they've been pushed to the sides, but we probably won't perceive going in and out of the iceberg scours. We just see them on the maps. So once we see an open sedimenty area, I think we're good to start some coring. Does Jeannie want to say hello? Third member of our team?
Oh, here we see a small jellyfish. Jellyfish swimming. We see also some corals. We see also some small crabs swimming in the water. There is a lot of what we call marine snow. This is particles that is coming from the sea surface, dead animals from the plankton, or fecal pellets, also from algae that are sedimenting, coming down to the bottom. And this is the source for the food for most of the animals that are living at the seabed. Here we see also on the sediment that there is a lot of greenish material, so this is all sedimented.
Oh, there's an
plankton.
octopus on the side of that rock.
Oh, that's a nice one, yeah.
Yeah. Should we zoom in and get some shots?
Maybe we should take a photo of this cephalopod, of this octopus.
So if we could get some glamour shots of that.
And on that rock there is also a lot of brittle stars. It looks really beautiful. It's amazing.
A collection of things with lots of arms. It really seems to be holding the body flat.
There is also a small fish.
There is a
Tom.
little ice fish, little notothenoid. Trying to act casual. There we go. Wow.
Great. And there's two small shrimps also near to the arms of the octopus.
There's quite a few. There's a little shrimp family associated.
Oh, yes.
There's one just under the brittle star arm, and then some little ones around the...
So it is amazing how these rocks are immediately colonized by organisms, huge diversity, many different species, and also very colorful. We have shrimps, we have what we call bryozoans, brachiopods, we have worms, we have brittle stars of different species. We have this octopus. Well, it's not the genus octopus, which is relative to it.
It's got a tiny, tiny shrimp just walking between its eyes.
Yeah.
There's a whole family of shrimp in here. Oh, I like these cameras. I could get used to this.
Yes, the quality of the cameras is amazing.
You can see what it's thinking. Are those, I think those are brachiopods, aren't they, on the...
Yes, the
base?
yellow ones. They look like...
I'm going to stop here since you have stopped too.
Oh, we got it.
They look like shells, but they are brachiopods. This is
Oh, there's a malted shrimp skin.
That's true. Coming down. This animal group, these brachiopods, they are ancient. They are known from many, many million years ago, from the fossil record.
Oh, mysids as well.
Mysids, yeah.
Okay. Did Sveta want the live brachiopods or only the dead? I think she just wants them for chemistry. Oh, no. Yes, she does want some brachiopods. Do you think we can collect those without upsetting the octopus?
That will be difficult.
Yeah, let's... Well, we're going to collect on the rock that's just behind anyway, so if they're in the area, hopefully we'll get lucky on that one as well. So just behind this rock, apparently, there is another one with one of our target sea cucumbers, holothurians, on.
Okay. Let's move to the...
I'm cautious not to land on the octopus, so maybe if we skirt around. Oh wait, oh, it's the one in the front? Oh, right there, okay. Thank you. I thought I was trying to see it in the background. Okay, so that brittle star is hugging the target holothurian.
Ah, that's the holothurian.
Yeah,
okay.
that's it. I'm glad she pointed it out because I'm struggling with these ones. They look like limpets.
Yes, I was thinking that's a limpet, indeed.
You want that? Oh, we're right here. Perfect. So we were successful with the slurp gun previously.
Yeah.
Because it's up on the rock, but they do tend to cling on. It takes a little bit of encouraging. But I know she wants them as low stress as possible. Do you have to remove the scoop to use the slurp?
So as you see, there are different tools.
Still learning.
Throw it in the basket now. You don't use it again.
There are different tools that we can use. This scoop. But there is also an additional tool that we can use to aspirate the animals or sediment or
Something
so
curled up drifting past.
It's like a vacuum cleaner, but for underwater. And then whatever we are aspirating will land in a small jar, which is on the other side of the ROV, and later on we can collect it from there.
Could you section brachiopods together with errants? There's loads of yellow brachiopods in that rock.
We can try. I don't want to upset the octopus. She might be brooding.
What? So let's see how it goes. The small British star already noticed that we are here, probably asking herself, what is this?
Yeah, it's on.
Well, actually we are in a place where probably nobody has been before.
Well done.
This area of the Bellinghausen Sea is very little explored.
What did you want off the bottom?
There's very little samples,
And if
very
that,
little biological
that rock
exploration.
just sort of centered to the left, the smaller one by the sponge, are we able to pick that up in a minute? So
That one right there.
yeah, if we can
Yeah, we should be able to.
go low impact on the octopus.
You want me to get some of these?
The yellow brachiopods if we can.
Yellow.
Yeah, I really don't want to disturb her if she is brooding.
Some on the left, Thomas, rather because he's
Yeah, is it closer on the... Basically these little shells, the yellow.
Yeah.
So this, this
A
octopus
few would be great, but it's mainly so we don't disturb the
breeding.
octopus.
maybe
so whichever
breeding
one,
the
depending
eggs,
on your
so we don't want to disturb it.
Yes, yeah, that's a good one. And then right at the base in the foreground, if we could grab the rock.
Oh wait, what jaw you wanted? Seven. Okay, well. That's in there. Okay, you can go for it. It's on.
Was the holothurian successfully in the jar?
Yeah.
Oh, excellent. Well done.
It's flock. 7, right?
Where's the time?
So we are trying to collect now these brachiopods.
Yeah.
They are known from the fossil record. Here it goes. Yeah, it was successful.
So what else you want?
And the rock
Try
in
to
the
get-
foreground. This little fella here. I think that's it for the suction.
Yeah, suction low. Come up with the
Thank you.
Oh, one of the crinoids has taken off. They're surprisingly mobile and they're beautiful and they swim. They sort of alternate the arms in two patterns and pedal their way up into the water.
America's taken the decision that we're going to have a bush coast.
So how many should we take?
Just for you, the geologist.
Okay, these are the 3 phosphate.
That version.
Okay, 3 for the geology.
Yeah, we get all those stuff that.
Yes. Sorry, thank you for reminding me.
You want to bring it up a little bit? So the darkness is the background. That's fine.
Yes, that'd be good. Give it contrast. Oh, we've got a free polychaete by the look of it.
Oh yeah, scour.
great. Can the minute rotate, or are we risking it then? Excellent. Horizons, all sorts on there. Great, thank you, team. Go from one of the bigger buyer boxes for that. Looks like the whole back row is free. Bible box 1. Yep, looks free. That's rock sample in buyer box 1. 1B.
1B or
Oh, it's no, sorry. It's just how it looks on my screen. No, go for either one. We only have, I think we're good. We'll hop on a little bit to give her some peace. Oh wait, sorry guys, hold on a second. Do we need sediment cores here?
Yes.
Like really specifically at this location?
No, we should move a little bit and go a little bit, find a place without rocks.
But I think Teresa needs one if we took the holothurian.
Okay, exactly. So maybe we take here one for Teresa. One push core.
Oh no, we. Oh, sorry guys, I'm getting confused. I don't think we do need them.
Okay. So we move to a place without.
Move to some clear sediment and just get the
Yes.
cores.
So then we will move to a place where we have nice sediment, and we will get the push cores there.
Sorry for the confusion, guys.
No, it's fine.
I think with only an hour left to go, we should prioritise the course. Well, it's my inconsistent bidding is what I worry about. It's my first time driving.
capture any squid on your squid cam?
I'm just reviewing the footage. There's certainly things buzzing around, but I haven't seen anything huge come into shot yet.
Okay, continue your transect, right?
Yes, yeah. So fully zoomed out lasers on. Thank you.
Bridge, are we? We can continue to move, please.
Moving on.
Thank you.
Is it a snail? Yep. Can we stop?
Okay. Yes,
Right.
we should try to get the snail.
RV bridge, if you just let me know when you guys stop, that reduces the chance that I accidentally run over you.
Stop now and you want us to.
Okay, we're stopping
Sorry,
again.
gang. It's just down here.
Well, I'll stop as well then.
So the snails are not so common. So it's good
There
if
it
we
is.
try to stop and get it.
Is that a snail? Or is it something coiled up? I think it's a coiled up fish.
Let's see, it's a little bit difficult to recognize at the moment. It could be a snail, could be a fish.
Could be a snailfish.
Fish.
Yeah. Maybe a zooacid? I think so. Could we have a little zoom? Oh, it's a blenny, I think.
Oh yes,
A
that's
zooacid, or it's not a snailfish.
That's a snail, a small fish.
Yeah. That's nice. I'm tempted, but I'm cautious that we do need to get these cores. So if we can carry on. Sorry, guys. It was really doing a good job looking like a snail.
Bridge, RV, we're on the move again.
Moving on.
What do you think of this patch coming up? Yeah. So this open patch just coming up. I think if we can set down and we'll hammer out the cores.
That's a slope.
Ah, right. Okay.
Okay, see how it's going down. So levels off to the left.
Good. Yes. It's
Yep.
It's kind of the top of the berm.
It's better if we move to a place where it's more flat. But we are almost there. We are
Oh, this is looking good.
at the margin of a iceberg core. Here now it looks much better. It's a much flat area. We can then land here and do the job. Take the bush course.
Hey Phil, we're setting down here to do some push cores, but I'm gonna let you come a little bit closer. We're still 100 meters off from the launch point.
Yeah, coming closer and then I'll stop once I have a delta of 50 or something like that.
Sure.
How many push cores are we doing?
We are doing 7. Oh, we're doing 14.
Okay. No, we haven't done any push cores.
It made it too long. What the geologists, I don't have the...
I thought they wanted 2 long ones. Yeah. Okay, 3 long. So there's.
Sorry, not delta. I meant RV to launch recovery point.
Yeah, that's fine. Oh, 50, 60, forever.
3 long and the rest of it.
So we'll do.
3 long ones. The rest of the regular ones.
So we'll get all 3 long ones right now.
Yeah.
That should be...
So we have cores from different sizes. For the geological
Yeah, 3, 5,
studies,
50,
3, 5, 6.
we need
3.
more sediment. We need cores that get deeper into the sediment. So we have some long cores
6 and 7.
And for the biological samples, we need smaller cores because the animals are living just in the first centimeters of the sediment. So we don't need that large, long cores.
Just a couple of centimeters of water.
So that's the first push core that we are getting in. Everything is soft sediment, so there is no problem. We don't have rocks beneath.
A couple of centimeters of water is enough.
This is good like this.
Is that okay?
Yeah, that's good. In total, we are taking 14 push cores here. This will take a while, but this will help us to characterize the environment. We want to know what is the grain size and also how much organic matter is in the sediment, which organisms are living there, but also what are the shells of little foraminifera and little mollusks that are living there. They can be used to reconstruct the climate in the past.
Great, thank you.
Very nice. The ones that are marked with colors, they are the long ones.
Did we not do that when we were taking the record? If that's how it's normally done.
Last time they would take a picture once we have all
Okay. Can we grab a screen grabber for free? And would you usually retrieve these before doing the smaller ones? Or lay them all out?
Yeah, I think we would, if there was like any special ones, we would recover those.
Yeah, if you
And
want
then
these
just-
for sure, then I'm gonna grab them now
Yeah.
so that they're done. Yeah.
Yep.
Okay.
So we are going to recover the long cores before we take the other ones. And let's see what happens. Yeah, very good. Now we go for the third one. Here it's coming out. Very nice. Perfect. Very good.
Grand.
So now
we have another
11.
Yeah, 11 standard.
Here we go.
You want them like close together, or how do you want?
Well, you can just enough of a gap so they don't disturb each other, but I think that can be within our reach.
Yep. That's good like this. Don't. That's good. Yeah. Leave a little bit of water on top.
Totally lost within that other push course.
The number of the last long push core. That was 3-6 for the first two.
3, 6, and 7.
7.
And 7.
3, 6, and
Cool. 7. And they're all along the front.
Yeah. They're where they were. So 3, 6, 7.
3, 6, 7. Okay.
So it goes 1, 2, 3, 4, 5, 6, 7. 6 and 7, 3.
Right. Okay, thanks. Thank you. It was quite a while ago. Anyone who's just joining us now, we've reached an iceberg scour mark, or at least an area full of iceberg scour marks, and we are collecting core samples. We've just deployed 3 of the longer cores, and now we're collecting 11 of the shorter cores for a mix of geology and biology. There's a lot you can do with the core. So the geo teams will be interested in the grain sizes and the makeup of the actual sediment itself and the history that it contains as it's been slowly deposited here. And then of course the biologists, it's full of life, and there'll be things that we haven't recorded in the past. As we're flying over this terrain, it feels like the dropstones that we pass are sort of oases of life, and that they contain a lot more life, and then there's this open sandy sediment. But that can be a little bit misleading, because actually it's that the life is in the sediment. There isn't much on the top. So it can feel like you suddenly get to these dropstones and suddenly life explodes, but there's probably just as much life here. It's just not as visible to us on the cameras. So that's why we have to then supplement that by getting these push cores. It's not as immediately rewarding. These will be taken back and peeked through with microscopes, but there is a huge abundance of life hidden within this sediment. You can see this sort of green tint to the surface, and you can see the cleaner areas where it's more of a lighter sandy color, and that is this food that is constantly raining down from above. You can see it in the water, this marine snow, and the water's surprisingly quite full of algae. The cameras we've sent down, there's quite a green tint to the water, and so all of that energy input is slowly raining down here to the deep sea. I think we're just over 600 meters deep, so not super deep, but we count as deep sea, and that's what's feeding this environment here.
In the sediments there are a lot of different species, but of course they are small. The animals, we can classify them in different groups. We have the megafauna, which are the animals that we can see here on the photo and video footage. They're large,
yes, it is 11.
Several centimeters or decimeters large, and we can identify them with photographs. Then we have a group that we call macrofauna, which they are around 1 centimeter large.
Yeah.
We need to use some...
Get a screenshot for the overall layout of the course.
Yes, we are going to take a screenshot. So the macrofauna is something intermediate, about 1 centimeter large. So we have the bivalves, we have snails, we have little crustaceans, crabs.
Yeah, excellent, thank you.
And then there are also smaller organisms that we call meiofauna. They are these organisms that they can pass through a sieve that is 1 millimeter mesh size. So they are quite small, smaller than 1 millimeter. But there is a lot of different species at this size. So there are little crustaceans, little crabs, there are also worms, there are water bears, there are mites. There are a lot of different organisms that are very small, and the diversity of them is very large. So specifically also here in Antarctica, this is some waters that are quite isolated, so most of the species that we find here of this small size, they are actually new to science. They have not been described, and we will be the first ones that have seen them. Nobody have seen them before. Now we are recovering the bush corals in these sediments. They are very soft sediments. We will then extract them from the corals when they are back to the vessel. And this is what we are going to use to find the small organisms, the small species that is in the sediment. In order to describe new species, we need to first do some microscopic slides. We need to consult the books, the catalogs, if the species have been found somewhere else in the world. And if not, we can make drawings, we can make photographs, we can use normal microscopes, but also electron microscopes or confocal microscopes that are using laser light in order to get images of the whole body, of the legs, of the different details. And it is the small details that is making also the difference between the species.
Yeah. So, do you think that's 13?
The Antarctica is as less explored as the deep sea. So here we are really expecting a lot of new species, and it is very important that we describe them, because the fauna that is characteristic for the Antarctica, this is what is making this continent so unique, that there are many species that are living only here, but we don't know them still. We still have a lot to explore.
It's a sea cucumber just parachuting in from the top.
Wow, great.
I see them take off, I don't often see them land. Not graceful.
By the way, Tom, what is the name of this fish that we have here?
The fish that keep bothering us?
Yes.
Right, those are... Now, I've been getting the name wrong a couple of times because I keep messing up my biblical J names. So it's the Antarctic Jonas fish, and they've been hanging around quite a bit with us. Actually, one of them went in the core tubes earlier, and we accidentally sampled one on the previous dive when it managed to get itself wedged into the tool tray. So they don't seem too fussed at all about the ROV. They seem a bit obvious when we're down here with this super silvery side, but we're adding a lot of artificial light here. And being this reflective, if you can't be transparent, being this reflective is a pretty good way of hiding yourself. So any sort of background bioluminescence, any sort of general twinkling in the water, you just become a mirror to that, and you don't cast an eclipse, you don't sort of cast a silhouette. When you're in the deep sea, casting a silhouette can be as bad as being visible yourself. And that can include the background bioluminescence as well. It's not just the light that's filtering down from above, it is the light that's just constantly going on within the water. It's hard to think about that actually, because we see things when we've got these lights on, but normally down here there will just be this background radiation, this background glow of bioluminescence. And where things make contact, where things are sort of rushing past a rock or stone or things like that, anything sort of disturbing that current, you get even more bioluminescence there. So you almost get this heat map of turbidity and disturbance. And you've got to sort of shield yourself from that. And a lot of these animals move in a very cautious way as well, because you're constantly setting off this light show by moving around. So maybe these guys, by undulating just that distant part of the tail, they keep their body quite rigid and just wiggle the tail. That's got to create a little bit of a light spot further down your body. So actually the fish part has gotten out the way before they're setting off that burglar alarm.
So now we have 4 cores to go.
Yeah. So that was our have to do. That's what would have gotten us in trouble if we ran out of time. So we can carry on doing a transect and going a little bit of going critter shopping and rock shopping.
Yep, that's good.
But we
there's still one of the holothurians that are needed.
Okay.
Let's see if we find one. They are normally. Oh no, I think we collected the 3rd one now on the rock, right?
I think that might have been the 3rd. Yeah, I'll double check.
That's true.
Yeah. While we're here, we do want to fire one more just before we finish. We'll just make a note of that last one.
This is the first place that we are sampling. We started sampling 2 days ago, actually. But we have on a map different locations that we want to visit. It's not being easy because of the ice conditions here in Antarctica. We need also always to look that the conditions are good for us to stay some hours and take the samples with the ROV on the same place. Of course, we are moving a little bit, but sometimes the ice is moving faster than the vessel, and we need to really take care that we don't have an accident with a big iceberg. So some of the areas that we wanted to go, they are really close by sea ice and also iceberg fields. And this is something that we need to look day by day. What is the situation, where the ice is, how the ice is moving, what are the weather conditions, and then we need to adapt and look for a place to do our research. It's now almost 1 past midnight, 1 in the morning, almost here. And today it is a wonderful day, very little clouds, a lot of sun, even at midnight. This is something that is strange to get used to, but we have always light. Here you see,
it should be.
You see the
It looked fire.
how much light and how much sun we have now. Midnight, 1 o'clock in the morning.
There it goes.
Okay, so we are done. We have collected the 11 push cores for biology, and now we can move on.
Thank you. Nicely done. So we have a hard curfew. We have to be home and in bed for 2, which means we have to leave the bottom at 1/4 the hour. So that gives us about 20 minutes to continue the transect and anything on our wish list that we'd like to sample. One of the most elusive things so far has been gastropods, so snails. Just a bit earlier we had a fish pretending to be a snail. False alarm.
So there was a question in the chat some time ago. It was about why do we need to collect the specimens anymore nowadays? It is really needed to take these specimens. There are different reasons why we are collecting the animals. One of the reasons is that the features that are needed to identify the species, they are in many of the animals microscopic, or let's say that you need to take a quick look, a careful look at the spines or the plates of the body, or the limbs, how many segments they have, and you cannot do this normally in photographs. And another reason is that we are also using genetic methods to characterize the species, but also to study the connectivity of the populations. And in order to use these genetic methods, we need to take a tissue sample and extract the DNA that we can compare them. For this, we need to collect the specimens.
Is the camera zoomed out a little bit? Yeah.
There is also another reason, that is that we are also studying the food webs. So we want to reconstruct how the food webs are structured, how diverse the food webs are. In generally speaking, who is eating whom and how it is the carbon flux through the food web in the Antarctica. In order to do so, we are studying what we call stable isotopes of carbon and nitrogen in the tissue of the organisms. This is telling us what is the position of the organism in the food web. For this, we also need to sample them. And then there is also another reason, that is we are collecting specimens for reference collections from different museums. In order to get an archive of life, we need to collect them. It's very useful to take photographs, but we need to have the physical specimens also in the collections.
One of the most difficult things is that a photograph is a finite point. It won't give you any new information. It can contain a huge amount of information, but it won't give you anything new. But our museum collections are continually providing us new things. Like we didn't have CT scanning, we didn't have genetic analysis just a couple of decades ago, and these collections that go back hundreds of years are now unlocking so much new data. So that's always the worry. We would like to go digital and rely on photographs and CT scans, but you never get any extra information, if that makes sense. It's a real finite point. So they're becoming tools that really help us, and digital holotypes are super helpful because, of course, these things are very fragile and precious, especially if there's only one in the world. And it's great to be able to send someone a file rather than sending them this precious singular specimen. But we do need that kind of anchor point for these species. We do need that kind of, here is an example of this species. And it's sort of an immovable object. It's like the kilogram and the standards that are locked away in Paris. It becomes the sort of definition of that species, and as these names move around and we change things, you need that fixed point. You need that this specimen represents this species. So that's the nature of a holotype. That's what it's doing for us. So we're entering one of the iceberg plough marks. This whole area has several iceberg plough marks. We probably won't see them at the kind of scale we're looking at. It's more something you see on the mapping, but we do have a fair slope. The seabed is slipping away to the right, so we might actually be on the side of one of these scour marks, following it along.
Iceberg scour marks, they are of course a disturbance to the seabed, but disturbance is not actually bad. Disturbance is helping to maintain a variety of habitats, a variety of conditions, and this is actually enhancing biodiversity. So we get more biodiversity in an area. Sometimes if there is more different types of habitats, and when the icebergs are passing by, they are producing, of course, a disturbance, but they are also helping in maintaining this habitat diversity.
Oh, there's something floating in the water.
Yeah, it should be a haddock nut. This is and now it's a jellyfish coming here. So now we see the
Is
bottom
that something on
topography.
the stem? So there's a, what's it? It's a sea pen, I think. Is it with the head closed? But there's something growing on the stem. Is that a sea anemone on the stem?
This maybe. Maybe we can take a photograph. And well, we have talked about the importance of getting the specimens and getting them in the collection, but of course we cannot deny that having a photograph of a living specimen is also extremely important, because
Stopping vessel, assuming you guys are stopping to.
yeah, I'm sorry, we are
doing a handbrake turn.
listening to the captain now.
Do you think we've covered it?
Because if we only have the specimens in the museum, normally they are kind of fixed in ethanol or in formalin, and they change their body shape. So it's very important to know how they look like in their true habitat, because this will help us to understand also how they live, how they feed, if they can swim, how they are using the environment, how they are using the sediment, if they are into the sediment or crawling on the sediment. So all these kind of details we can only get if we are really diving with an ROV or with towed cameras, but of course if we are getting footage, videos, photographs, which color the animals have.
Yeah, so is that...
Yeah,
I think that's a sea pen with a...
Yeah, that's kind of
anemone on it.
Anemone, yeah, growing on a sea pen, yes.
I'm guessing it was smaller when it got on there.
Yeah. So here you see a good example that for all the animals that are filter feeders, like these anemones or the crinoids or other animals that we have seen, it is very convenient if they get a little bit off the bottom. So if they can crawl on stones, if they can crawl on sponges, or if they can crawl on, in this case, like a sea pen, so they get a little, some centimeters or some space from off the bottom, and they get much better access to the food that is coming from the water. So specifically for filter feeders, it is good to get a little bit higher and crawl on the structures that are there. This is the reason that these dropstones that we can see once in a while here, they are immediately colonized by lots of species. And they are very important in maintaining also the biodiversity in the area.
When you were saying earlier about disturbance being a good thing, it's where we get some of the highest diversity. There's kind of a sweet spot. So if things are too stable, dominant species sort of set up and outcompete everything else. If things are too dynamic, then you just end up with the sort of extremophiles, the really sort of tough early pioneering species. And there's a sweet spot in between with just enough things messing things up and reshaking the dice every now and then. And that's where you end up with a lot of diversity. So these iceberg scour marks are a great example of that. And they could be hundreds, maybe even thousands of years old. This is a really stable environment. So these plow marks are going through and they might hang around for hundreds of years. But that is going to have different impacts on the communities that we find there.
Yeah, we see a very nice sea urchin.
The jellies.
Jellyfish. Sea cucumbers, 2 different species. They can get very abundant sea cucumbers sometimes.
They're really strange to hold. They feel like water balloons. You can see the gut inside them, but their organs are pretty free and easy in there, and it feels like a snow globe. It feels just like this bag filled with fluid, and you can see it swirling inside them.
Yes, they always remember me to cows on the fields, the
grazing.
They're grazing there, sitting there, staying there and sitting like it would be cows. And indeed, these sea cucumbers are one of the first that take profit of the fresh algae that are coming from the water column and sedimenting on the sediment. All this greenish fluffy thing that you can see on the sediment. And the sea cucumbers, they are grazing, they are feeding on that, and they are one of the first organisms that take profit of this. They are also called sea pigs, but I think that more convenient would be to call them sea cows.
Can you make out what the orange thing is? So anyone just joining us, we're following along an area of iceberg scour marks. So some of these icebergs are so big that they actually make contact with the seabed and plow these marks in the seabed. And they hang around for a long time. Some of these might be hundreds of years old, and you can kind of figure out, a little polychaete swimming by the camera, you can kind of figure out which is the youngest and oldest by which one sort of passes over the other one. But it's tricky to figure out just when all that happened.
There's some nice sponge also here on the rock.
Oh yeah, I missed that one.
Sponges are very common here in Antarctica.
I'm seeing some huge glass sponges.
And they grow very, very tall. They are very big. They can be 1.5 meters large. These are glass sponges, most of them. Here we see also a sea star.
What do you think of the smell of a glass sponge? So this is the thing, there's people watching at home don't. It's a distinctive smell, isn't it?
It is.
It's like an ammonia chemically smell.
Yeah.
And the spicules are horrible.
Yeah, it needs, you need to take care if you want to touch one of them, because you will end up with a lot of spicules in your fingers.
Yeah. Tiny filaments of glass that that really get into your skin and irritate it. And you feel fine at the time. You can be playing in the mud and having a great time, and then you try and go to sleep that night and suddenly your hands are burning, and it's all these nasty little spicules. So certainly gloves when you handle a glass sponge.
So why do you think that this fish is at the bottom? It is more safe to be at the bottom than to be in the water?
Well, most of the ones we see chilling out on the bottom are the plunder fish.
Okay.
So they are sit and wait predators, and they've actually got a little fleshy lure on their bottom lip. So like the anglerfish have it coming out the top of their head, these guys have a little fleshy lure coming out the bottom of their lip. It's not quite as sort of evolved as the anglerfish, which seem to pretend to be things like squid and copepods and really specific things. It tends to be sort of a fleshy lure, and part of that is that they often accidentally eat them or bite bits off them when they snap at their prey. So they're sit and wait predators, and they'll wiggle this little lure on their bottom lip to try and entice things in.
So there is a question in the chat if we are under the ice.
This is an area that is under ice for a lot of the year, and that's why we're so excited to be here. People don't usually get a chance to see here.
Yes. But the ROV is now not under the ice. We are really searching for places that are ice-free at the moment, so that we can dive and stay for several hours in a place and move. We are making something like between half a mile and a mile during these dives, and it will be very dangerous if there will be ice at the bottom, at the surface. The vessel is following the ROV. If we have icebergs or ice approaching the vessel, then the vessel needs to move and the ROV needs to follow. At the moment we are doing a video transect. This footage can be analyzed later. We will then count how many organisms we have per square meters and which is the diversity of the different organisms. And we are then sampling some of the organisms that we find they are interesting or that different scientists want to have. But we are approaching the end of the dive, and we have collected several rocks and a lot of sediments with the push cores, and we have collected some corals. But now we will end the dive in a couple of minutes and go back to the surface.
Okay, I think that's a wrap. So that concludes our dive. Thank you everyone who's joined us for this. We're going to start our ascent soon to make sure that we're on the surface in time to move on to the next side.
It will take about 1 hour for the ROV to come up to the surface, and then the work starts for us. We will then now to process the samples, label everything, make the catalogs, put everything in databases, and pack everything to get it back home to the research institutes and the different museums.
Yeah, it's all fun. Process all this. I think we leave the stream on the way up. I'm not sure, but if we do, there's always the chance for some interesting pelagics. Lower chance of success. It's a pretty open area up there, but it's pretty exciting when we do see something. I know there's a few folk in the chat hoping for some squid on the way up.
Yes, we all hope to see squids. Hope also to find the colossal squid here.
Take one home as a pet.
Let's see.
Someone in the chat is asking if the samples have to be kept under pressure. There are some high pressure samplers out there, but there are tricky things to use because essentially once you bring them up you've got a bit of a bomb. So we allow them to equilibrate with ambient pressure. That can sometimes be tricky. When you're working with super deep sea stuff, actually at the molecular level, the molecules that make up these animals are adapted to that pressure, and they start breaking down once you bring them to the surface. So I work a lot with Hadal snailfish, and they are made of like this gel, and they actually break down at atmospheric pressure. The biggest issue for us here is the temperature. Again, these things warming up, they start to degrade really, really quickly. So there's actually a cold room on board, and it's a little bit chilly work, but a lot of this work will be done actually in a cold room to try and maintain the specimens for as long as possible. I know those holothurians, the sea cucumbers, are going to be investigated in there because Teresa's doing some feeding experiments with them, and she wants them as undisturbed as possible, so she's going to try and make them happy in the cold room.
Thank you. We're going to start at 10 o'clock.
Thank you.
We have liftoff.
What time are you expecting the first floats?
Here we go.
Yeah, likely in about half hour.
Half hour to float.
Coming back home.
So.
Thanks everyone who joined us for this dive. It's been great having you along, actually. It's my first time with a dive stream, and it's been nice seeing the chat and seeing, I don't know, feel like you're all here with us. So thanks again for joining us.